lineage. Basically, in humans, there will be two types of cell lineage. One will be the somatic, and another will be germ cell. See, if you talk about the somatic germ cell lineage, then surely concentrate on the current generation. For example, if a cell is there, damage is happened, it need to be repaired. It is under the control of somatic cell lineage. And one more example, if cell is there, it is increased in terms of the cytoplasm, then it is also under the control of somatic cell lineage. Now, on the other hand, if you talk about the germ cell lineage, it always concentrates on the future generation. How they concentrate on the future generation means by creating the offspring. How this offspring will be created? By the union of gametes. And the process that the gametes are produced is known as gametogenesis. See, when we talk about the gametogenesis based on the gender, it is of two types. If it is happening in the female, then it is oogenesis, where the female gamete is produced. And if you talk about the males, then it is known as spermatogenesis. It is the process that produces the male gamete, nothing but sperm, along with seminal fluid. Okay, these are the basic cell lineage present in the humans. Now, let us concentrate on the particular topic, nothing but spermatogenesis. That means production of male gametes and the release of male gametes from the somatic mother cells is known as the spermatogenesis. So, first, we talk about what is meant by spermatogenesis and what are the structures that are involved and all those things. Okay, let us see. These are the contents that I will be discussing in this topic. First, I will talk about the structures of the testes, seminiferous tubules. Without having the proper knowledge on the testes and seminiferous tubules, we can solve the problems and we can solve the questions that obtained from the based on the location wise. That means there will be basal and the central part of the seminiferous tubule, from which area the spermatogonia will be present, and in which area the primary spermatoid will short meiosis. Like those questions, we need to be solved. Surely, we need to be thorough with the structure of these testes and seminiferous tubules. And the second subtopic is spermatogenesis and technology that are involved in this process. Will be clearly discussed in those second subunit. And the third one is spermatogenesis chart. It is the simplified one, simplified into diagrammatic chart. If you are thorough with this chart, then whatever may be the question based on the ploidy or based on the number of chromosomes or number of chromatids and from where it is division is continued, all those things will be clearly mentioned and it is written by me in a short notation. Next, fourth one, the factors that are involved and that monitors this spermatogenesis, either it may be physical or environmental, whatever it may be, those will be covered under the fourth subtopic. Next, firstly, we will talk about the testes. Before that, let us take a slide, new slide. See, first, testes will be there. The testes, basically, when we talking about the spermatogenesis, we kept the characteristics of testes, all those things aside. Just I talking about the coverings, and the remaining parts will be covered when I open with when I go on with the slide. Okay, testes, just coverings. Testes always covered with three layers of coverings. From outer to inner. If you talk about outer to inner, the outermost layer is tunica, vaginalis, and middle layer is tunica albuginea, and the innermost layer is tunica vascularis. Okay, first we will talk about each layer, then we will go into the topic. See, if I talk peculiarly about tunica vaginalis, it is divided into two, again divided into two layers, outer and inner. Outer one is parietal layer, and inner one is visceral layer. A point to be noted here that in between these two layers there will be fluid. This fluid is known as hydrocele. Hydrocele. If you talk about the components of hydrocele, nothing but majority, 99% will be the water plus solutes. Solute may concentrate. If you talk about the solutes, there will be electrolytes, proteins, small amount of proteins. Okay, these are the things. See, the scrotum maintains always 2 to 2.5 degrees Celsius less temperature than the body temperature. Why this is? Because of this hydrocele. Presence of this hydrocele, the scrotum maintains the temperature in testes that should be 2 to 2.5 degrees Celsius lesser than the human body temperature. Okay. Next, if you talk about the middle layer, it is the most important layer because it forms septa and divides the testes into testicular lobules around 200 to 300. The range may be varied from book to book, and average it will be around 250 testicular lobules. Okay, based on this layer, that means tunica albuginea, the whole spermatogenesis will be evolved. Next, if you talk about the third most important layer, tunica vascularis, it is the layer that produces nutrition to the testes layers. Why? Because see, tunica vaginalis, tunica albuginea, they are only the layers without capillaries, blood capillaries, without blood capillaries. But tunica vascularis is the layer which possesses blood capillaries. Because of these blood capillaries, nutrition is only given by these testicular vascularis to the other two remaining layers of the testes. See, covering these all three layers of the testes, there will be one more layer. This layer is filled with muscles, nothing but cremaster muscles. Plays major role in maintaining the elevation of testes. Keep it in mind, elevation of testes. That means if temperature is very high, then cremaster muscles will become non-contraction, and it will lead to the lowering of these testes. Okay, if temperature is very low, then it will be contracted and cause the contraction of these cremaster muscles and cause the upward movement of the testes. Okay, next, and one more layer surrounding this cremaster layer, that means cremaster muscles, there will be one more layer. It is the peritoneal layer that covers the entire testes, and this layer is called as externa spermatic fascia. Okay, after outer to this, there will be one more layer. That layer is Daughter's muscle. It is made up of muscles which cause the elevation of scrotum. Keep it in mind, cremaster muscles involves in the elevation of the testes, and the Daughter's muscles involves in the elevation of the scrotum. Okay, keep it in mind. Both the cremaster and Daughter's muscles combinedly cause the upward and downward movement based on the temperature present in the scrotum, along with the testes. If the temperature is high, one type of mechanism will be happen. If temperature is low, then other type of mechanism will be happen. Those will be discussed in further slides. This is the coverings. Now, okay, as I informed you that testes, if you talk about the characteristics of testes, number of testes will be two. There will be two testes separated by the septa scroti. And if color is pinkish and shape will be oval shape, and position outside the abdominal cavity within the pouch, those pouch is known as scrotum. This scrotum maintains the temperature that is required for the process of spermatogenesis. That means 2 to 2.5 degrees Celsius lower than the body temperature. Next, descendancy time. It will be around 7th month of the gestation, and the passage through which this testis will be descended into the scrotum, those canal is known as nothing but inguinal canal. And if you talk about the function of the testis, purely it acts as the primary sex organ, and also it acts as the heterocline gland. How it acts as the heterocline gland? If we talk about that, then heterocline gland means then it is perform the mixed function, both exocrine and endocrine. Exocrine and endocrine. Exocrine, secretions. That means production of sperms. Involves in the production of sperms. Endocrine, production of hormones required for sperm production. Example, inhibin, anti-mullerian hormone. Not only sperm production, for also sex differentiation. Both sperm production and sex differentiation. For those, what are the hormones required will be produced by these testes because of presence of SRY gene. Okay, keep it in mind. So because of that, both exocrine and endocrine function, this gland is called as the heterocrine gland. If you talk about the covering, there is 3 layers. Already I discussed all these things. Tunica vaginalis, tunica albuginea, tunica vascularis, and tunica albuginea is the most important one because it causes this separations in the testes. Okay. Now, see, this is the structure of the testes from outer to inside. If you talk, outer skin will be present, Daughter's muscles will be present. After that, external spermatic fascia will be present. After that, cremaster, then tunica vaginalis. Tunica vaginalis is also divided into 2 types. Outer layer is outer tunica vaginalis is nothing but a parietal layer, and the innermost one of tunica vaginalis is a visceral layer, and inside that there will be tunica albuginea. Inside that there will be tunica vascularis. See, I will enlarge the peak. You can clearly see this. This is the structure. See, this is the tunica vaginalis outer layer, that means parietal layer, and the inside layer is this one. In between these, there will be fluid, nothing but hydrocele. Okay, tunica albuginea, this where the separations happened. Okay, this is the tunica albuginea, which divides the testis into testicular lobules. It is one of the testicular lobule. It is also one testicular lobule. It is also one testicular lobule. Like this, these are the testicular lobules. Okay. Now, okay. Before that, one more slide. I will take one more slide and I will. Okay. Now, tunica albuginea, which cause the division of testis into testicular lobules. How many will be there? Around 250 testicular lobules will be there. Each testicular lobules, like this 250 will be there. Each testicular lobules consist of 1 to 3. That means 2 to 3, not 1 to 3. Seminiferous tubule plus seminiferous tubule plus seminiferous tubule plus intertubular space. See, the testis, if you talk about one testis, there will be two in number. I particularly talking about only one testis. When one testis is separated by the tunica albuginea, then it is divided into 250 lobules, nothing but testicular lobules. These are testicular lobules. Okay. Then each testicular lobule is divided into 3 seminiferous tubules plus the space between these. See, if you talk about these are seminiferous tubules and outer one is the testicular lobules. The space between 2 seminiferous tubules is known as intertubular space. Basically, during the origin of, that means gestation period and before the puberty, it is filled with mesoderm, from which the Leydig cells will be originated. Other than these Leydig cells, two more types of cells will be present. It is one, second, third one. Nothing but immunocompetent cells, which cause immunological functions. These immunocompetent cells are microblast cells and fibrinogen cells. Okay. Other than this, there will be blood capillaries. Okay. Now, if you talk peculiarly of the seminiferous tubule structure, outer to inner. Outermost layer is there will be PMC, peritubular myoid cells. After that, basement membrane. After that, seminiferous epithelium, which possesses. See, this seminiferous epithelium is divided into two types. That means produce two types of cells. One somatic cells, and second one will be the spermatogonial germ cells. Okay, these somatic cells nothing but Sertoli cells. Spermatogonial germ cells is nothing but spermatogonia. Okay, these are the outline of the what are the structures that present based on the separation of tunica albuginea. I will show the diagrams for this, and I will discuss those with what are the slides prepared by me. See, testis. I already informed you. Each testis is divided into testicular lobules. Around 250 testicular lobules will be present. Okay, it is due to the layer, the middle layer of the testis, nothing but tunica albuginea. Okay, keep it in mind. One testicular lobule is composed of 2 to 3 seminiferous tubules and the blood vessels, interstitial cells, nothing but Leydig cells, and immunocompetent cells. Okay, if you talk about the LC, those are the structures, that means cells that produce androgens. They are the main hormones that involves in the major functions, which will be discussed in further slides. And the chief androgen that will be the major and the chief androgen that's secreted in the human, especially in males or females, it will be testosterone. One more fact is that testosterone will be produced both males and in females. Okay, but the transmission into DMT, DHT, not DMT, DHT, dihydroxy testosterone. This lead to, and from where testosterone is also converted into estrogen. In females, it's happening, and the testosterone converts into DHT in males. Because of those, the sex differentiation is involved. It will be discussed in further slides. Next, IMC, nothing but immunocompetent cells, fibrinogens and macrophages. Already I discussed these things. And mesoderm around the seminiferous tubules forms the interstitials. Already I informed this. Next, if you talk about if you see the structure, see, it is the testis. These are all testis. Okay. Testis is divided into testicular lobules. Whatever the thing that we can observe here, it is the testicular lobules. Each testicular lobule contains here one structure like this. It is the seminiferous tubules. There will be 2 to 3 seminiferous tubules in each testicular lobule. These are the inside. There will be like this. Nothing but those are the structures, sterol cells, and all those things will be present inside the seminiferous tubules. See, it is one seminiferous tubule, and it is another seminiferous tubules. The space between these seminiferous tubules and these seminiferous tubules is called as the intertubular space. This space is filled with the LC, Leydig cells, blood vessels, and other than these two, there will be fibrinogen and macroblasts. Okay. Next, seminiferous tubules. Now, seminiferous tubules. These are the highly coiled tubes. Each tube is lined up by 2 types of cells. Already informed. It is the somatic cells, actual germ cells, spermatogonia, and these 2 cells will be rested in the stem cells niche. Okay, keep it in mind. What is meant by stem cells niche? Let us discuss about that. Stem cell niche. I will enlarge this a little bit. Stem cell niche. See, stem cell niche is the junction that is created by 1. Leydig cells, 2. Vascular endothelial cells, nothing but blood capillaries. Third one, immunocompetent cells, immunocompetent IMC, immunocompetent cells. Next, 4th one, Sertoli cells, somatic cells of seminiferous tubule. Somatic cells of seminiferous tubule. Very, very important. 5th one, see, spermatogonial stem cells. Stem cells. Actually, these 4 will cause a junction. See here, it is one, it is another cell, it is also another type of cell, it is also another type of cells. These cells cause one junction near the epithelium, that means PMC, near the PMC, and this junction is known as somatic stem cell niche. Okay, in that stem cell niche, those germ cells, that means spermatogonial stem cells will be reside. Okay, keep it in. Now, see, this is the structure of. I will show the structure of seminiferous. See, from outer to inner, if you talk about the structure of the seminiferous tubules, they will be PMC. In some books, it is referred as PTC, that means peritubular myoid cells. Okay. Next, inside that there will be one basement membrane cover above this PMC. There will be one basement membrane. Above that, there will be seminiferous epithelium. These are divided into 2 Sertoli cells and spermatogonial germ cells or spermatogonia. See, these are the spermatogonia cells and these are the Sertoli cells. Sertoli cells. It is the lumen inside. Okay, keep it in. Next, see, this is the structure where you can see the clear line. If you talk about the testicular lobules, this whole structure will be the seminiferous tubule. And another seminiferous tubule, let us think that there will be another seminiferous tubule. In between these 2, there will be in a intertubular space. In the space, there is Leydig cells. These are the Leydig cells. And it is the venule which carries the deoxygenated blood. And it is the arteriole which delivers the nutritionless blood, that means oxygenated blood. And it is one seminiferous tubule. If you talk specially about this seminiferous tubule, I will enlarge it. See, this is the seminiferous tubule. Here to here, whatever the space. Okay, it is the space which contains the lymphatic channel or sinusoids will be present. Lymphatic sinusoids will be present. And it is the wall of the seminiferous tubules which is made of the fibroblast and myoid cells. And inside it, there will be seminiferous epithelium, somatic cells, nothing but Sertoli cells, and spermatogenic cells will be present. These are the spermatogenic cells, that means spermatogonia, spermatocytes, spermat, and from outer to inner. The spermatogonia will be in a basal compartment, and these two will be in the central compartment. What is this basal and central compartment? I will discuss in the further slides. Okay. See, these are the spermatogonial cells, and there will be the setoid cells. These are the setoid cells. Okay. Next, see here. It is the one seminiferous tubule. It is another seminiferous tubule. It is one more cell. There will be lymphatic sinusoid, as I discussed here. There will be intertubular space, which is filled with lytic cells, arterial, and all those things. Okay. If we see the specifically this, it is the stem cell nature, which is a junction of those already discussed 4 types of cells will cause the junction, and those junction will be the stem cell nature, where the spermatogenic cells will be present. Okay. Next. Oh, just keep it in mind. Seminiferous tubules. See, based on puberty line, that means the time of the puberty, before the time of the puberty, before and after. Basically, seminiferous tubule, before the puberty, they are called as sex spots. They're actually called as sex spots. And after the puberty, it is called as the seminiferous tubules. What is the difference between the sex spots and the seminiferous tubules? Only these things. That means, see, if I talk about the sex spots, it contains outer to inner. If I talk, there will be PMC, peritubular myelin cells will be present. Basement membrane will also be present, and inside that, seminiferous epithelium will be present. Of course, it divided into 2 types: setoid cells and spermatogonial stem cells. See, these spermatogonial stem cells will not be there. Instead of these spermatogonial stem cells, there will be PGC. That means primordial germ cells will be present. What is meant by primordial germ cells, and what the transformation from primordial germ cells to stems, and the stages will be discussed in the further slide. So first, just to see the difference between these seminiferous tubule structure before the puberty and after the puberty. Okay. Inside that there will be no lumen. Lumen will be absent. What the difference? First one is lumen will not be absent, will not be present, and second one, instead of SSC there will be PGC. That means primordial germ cells will be present. See here, inside this there is no lumen. Lumen is absent. Just before the puberty, what will be the morphological changes that occur in the seminiferous tubules means that development of the lumen, and second one, the transformation of the PGC, that means primordial germ cells, into spermatogonial stem cells. PGC into spermatogonial stem cells. Just before the puberty, these things will be happened inside this sex pod, and these sex pods, after developing of these morphological structures, then it is called the seminiferous tubules. That means now what will be the structure of the seminiferous tubules after the puberty? Then it will be outer to inner. PMC will be present. Basement membrane will be present. Epithelium, that means seminiferous epithelium will be present. Inside, lumen will be present. Lumen present. In seminiferous epithelium, setoid cells is common. Setoid cells is common. But instead of PGC, the PGC is developed into SSC. That means spermatogonial stem cells. Keep it in mind. These are the 2 morphological structures that we can see in before the puberty and after the puberty in the structure of these seminiferous tubules. Okay. Next. See, before that, I will take one new slide and I will go with the stages of the cells. See, stages of germ cells. The 2 male germ cells. Okay, keep it in mind. Now, we can say that male germ cells that are happening in the testes. 1 minute. That are happening in the testes or male gonads. Okay. Why I remove the male germ cells? Because there will be one peculiar reason for that, and I will discuss when after completion of these phases. First, there will be PGC, primordial germ cells, okay, which is developed from the epiblast layer. That too peculiarly, the epiblast is divided into 3 major separate type of tissues, and from that endoderm will be developed. From this endoderm, if we talk about peculiarly, from this endoderm there will be production of PGC, and which PGC after migration and reach the gonad, where they are called as gonocytes. PGC will be called as gonocytes when they reach the gonads. Now these gonads will be converted into spermatogonial stem cells. These spermatogonial stem cells will be converted into spermatogonia. Okay, spermatogonia is again divided into type A and type B. Keep it in mind. Type A is again divided into A single, A pair, A aligned. That means type A follows 4 to 6 mitotic divisions and cause the development of these 3 types of cells. Okay. Next, type B. Spermatogonial type B will be present, from where primary spermatocytes will be present, will be developed. from which secondary spermatocytes will be developed, from which spermatid will be seen. Final structure sperm will be or spermatozoa or spermatozoon. In some books, they given as spermatozoon, ZOON. Okay, this is the hierarchy that involved in the stages of germ cells that based on the timings. Okay, see first, if we talk about peculiarly about PGC, that means primordial germ cells. What we will be discussing first, where it will form, formation. Second one, migration. Third one, characteristics. And fourth one, abnormality. This will be discussed in the slides. Okay. PGC. During the second week of gestation, the PGC will be developed from the epiblast, particularly from the endoderm. Okay, and these PGC which are formed will now they need to be shifted to gonads. Okay, they need to shift from their origin, that means from the site of the endoderm near the yolk sac. Near the yolk sac, see here, PGC. It is the yolk sac. Just to the junction between allantoins and the yolk sac, here from the epithelial wall of the yolk sac, there is the development of PGC. Okay, now this PGC need to reach the gonadal ridge, which is the site for the development of the gonads. Okay, see what will be the pathway, and the timing is also different. First, we will talk about the timing, then I will talk about the migration pathway. Okay, if we talk about the timings, when this migration and which week this migration will be completed and it will be started. See, in second week, formation will be seen, and the movement will be started within the second week from the wall of the yolk sac towards the gonads, and in fourth week, migration starts to developing gonads, and end of the fifth week arrives to the developing gonads. Okay, that means by the end of the fifth week of gestation, it reaches the gonads. And keep it in mind, while migration, that means from moving from yolk sac to gonads, there will be happening of mitotic divisions and increase their number. Okay. Next, if you talk about the conversion of PGC into SSC, this just happens just before the puberty. I discussed this thing when I given clarity on the difference between the sex cord and the seminiferous tubule. Okay, keep it in mind. Next, if I talk about the peculiarly about the migration pathway. Okay, here see. These are the primordial germ cells formed near the wall of the yolk sac, just to adjacent to the allantoins. These are the allantoins. Just to adjacent to the allantoin, from the wall of the yolk sac, there will be development of the PGC. Now PGC need to shift towards the gonadal ridge. This is the gonadal ridge where the gonads are present. The pathway. If I talk about the pathway, first the PGC in yolk sac, they reach the hindgut. From hindgut to dorsal mesentery, DM, from where it reaches the gonadal ridge, which is the site for the gonads. Okay, this is the pathway. This whole pathway starts in the fourth week. PGC formation happened in the second week, and by the end of the fifth week, this migration is completed. Okay, keep it in mind. This is the pathway. Okay, now see, as we discussed one thing, that while migration, the PGC should migrate from yolk sac to gonads. There is one abnormality. What that abnormality? In case if they not reach the gonadal ridge or gonads, and it reaches the extra gonadal space, then what will happen? Generally, the cells that reaches the extra gonadal space, that means this is the gonadal region. Other than this, they reaches from while migration, they reach from this region. They not reach this region, and they reach into some other regions like kidney tubules or nephrons or something, whatever it may be. Then usually they will die. First cases, usually they will die. If not, they not died, then the condition is known as tetratomous condition. Okay. Now let us discuss about what is meant by tetratomous discussion abnormality. See, tetratomous. What is meant by tetratomous? That means accumulation of PGC other than the gonad, the ridge region. Then this abnormality will be caused. Okay, that first condition. Second one, it is the mass of undifferentiated tissues like skin, nails, hairs, etc. Okay, keep it in mind. Tetratomas is a condition in which the PGC, usually which are need to be migrated to the gonadal region, is not happened and develops an abnormality of mass of undifferentiated tissue accumulation. This undifferentiated tissue is due to the accumulation of skin, nails, and hairs. Okay. Types of tetratomas. Based on the region, the PGC is shifted and accumulated. That means this all things comes and accumulated. If it is happening in a mediastinum, then it is mediastinum tetratomas. If it is sacrocochial region, okay, then it will be the sacrocochial tetratomas region, and the oral region, then it will be like oral tetratomas abnormality. I will show the diagrams. See, here is it is the sacrococcygeal. That means here the baby will be like this. Here the head region will be present. It is the placenta. It is the limbs part. Here the sacrum and coccyx will be present. Here sacrum and coccygeal region of the vertebrae will be present, where the accumulation of these undifferentiated tissues like the skin. If you cut this one, then we will see the skin, hair, etc. Okay, this is the oral lesion. Actually, there will be development of oral lesion, but due to this condition, the accumulation is happened. Okay, next. If you talk about the pathway, already I mentioned that PGC will be converted into SSC, which will be differentiated into spermatogonia, and which will be the primary spermatids, secondary spermatids, and after that, a spermatozoon will be developed from by the morphological differences happened within the spermatids. Okay, next. See, if you talk about first, we discussed about the timing. Second one, we discussed about the migration pathway. Third one, abnormality. Fourth one, characteristics. See, it is the main area we need to be concentrated, because up to now what we think will be totally different, and it is the new concept that we are supposed to be learned. Characteristics. See, when we talk about the characteristic of PGC, firstly, it is bipotential. Why we call this bipotential cells? Because they have the capability to produce both male gametes, nothing but sperms, or female gametes, nothing but ovum. Okay, either of these two gametes will be produced, not both, either of them. Which one will be produced? Those things will be determined by the gonad in which they are migrated. If the gonad is testis, it will produce one type of signals, and those signals cause these PGC cells to develop the male gametes. Okay, if the gonad is not testis, and if gonad instead of testis they reach the ovary, then surely there will be development of the female gamete. Okay, now what causes this development of the male gamete whenever it reaches the testis? Because the testis gives these signals which cause the production of the particular gamete, nothing but sperm. Okay, what those signals? Those signals will be have profound difference between spermatogenesis and oogenesis. If you talk about peculiarly one type of difference, it is timing of meiosis. One and most important difference that developed due to because of these signals produced by the gonads is timing of meiosis. See, in females at embryonic gonads, this meiosis will be started and arrested at prophase 1, meiosis 1, prophase 1, diplotin 1. Okay, but in males after puberty, the meiosis will start. What makes this difference of timing when we talk about the meiosis and the appearance of the meiosis? Why this timing is different? Let us discuss about that. Before that, I will take one new slide and I will give you clarity. See, PGC migration happened, reach the gonads. If it is in ovary, if it is in testis. Okay, keep it in mind. If it reach the ovary, okay, the development of the female gamete will be seen. If it reach the testes, it produces the sperm, nothing but male gamete. Okay, in terms of we are discussing about the timing of meiosis. So timing of meiosis. Okay, in females, it starts in gestation itself. It occurs after puberty. What makes this difference? Okay, first we will talk about the ovary, then we will talk about the testis. See, in ovary, upper regulation of STRA8 enzyme. It is not enzyme, it is gene. Okay, upper regulation of STRA8 gene will be seen. Where in testis, it will be down regulation of STRA8 gene will be seen. Okay, what are the major difference that causing the variation in the timing of meiosis in ovary and in testis means because due to this STRA8 gene, which is the gatekeeper of meiosis. Okay, what are the causes that causing the upregulation and downregulation of this STRA8 gene? Let us discuss. See, WNT will be present, and one more thing, retinoic acid will be present. Okay, these two cause the upregulation of STRA8 in ovaries in gestation period itself. See, RA increased, cause the STRA8 enzyme gene concentration. It is gene. Okay, it acts as the transcription factor. That's promote the meiosis. These all things happened in gestation itself. Gestation. Okay. Now meiosis will be arrested after certain time period. It will be arrested in meiosis 1, prophase 1, at diplotin 1. Okay. These things will be clearly mentioned when I deal with the oogenesis. Okay. This is the pathway. If we talk about the testis, the totally different when we compare with the ovary and the testis. The mechanism involved in the ovary and the testis. I talking in testis. First, 13.5 days. After 13.5 days to puberty. After puberty. Okay. 13.5 days. After 13.5 days to puberty. It is a special case. And after puberty, there will be 3 stages based on the timings. See, retinoic acid will be present in testis, which will be secreted from the mesonephrons of kidneys. These produce the retinoic acid, and retinoic acid will cause the development of the gene STRA8. But what the difference is, here there will be one protein called Cry26B1. Okay, these are the enzyme that cause the inhibition of this conversion of retinoic acid into STRA8. Okay, because of this protein, the conversion of retinoic acid into STRA8 is happening, thus from causing the arrest of meiosis until 13.5 days. From first day to 13.5 days, it causes the arrest of meiosis. Okay, after 13.5 days, what will happen? Now the retinoic acid will be present, which can be converted into STRA8. That means gene of the STRA8 cause increase because the protein that are involving here, these concentration will be gradually decreased after 13.5 days. Gradually it will be decreased. That means there is no inhibiting protein. Because of lack of this inhibiting protein, the retinoic is totally converting into STRA8 gene, which will act as the transcription factor for meiosis. Even though meiosis will not happen. Why? Because the inhibitor of this gene, nothing but nanos will be present. It causes the STRA8 enzyme suppression. Because of these, from the day of 13.5 days to puberty, the conversion of STRA8, the function of the STRA8 enzyme, that means initiation of the meiosis and the promotion of the meiosis will not be happened in the testis. Because of this, meiosis will be stopped. Okay. Now after puberty, what will be the situation? Now retinoic acid will be present. First two cases will be present. Retinoic acid is present. There is no inhibited protein that inhibit the conversion of retinoic acid into STRA8. So the STRA8 gene will be concentration will be increased, and there will be no nanos after puberty. So the conversion the function of the STRA8 gene can be accomplished, and the meiosis promotion will be started. Okay, it is one of the things. Second thing, there is the development of GDNF, SCF, and other paracrine functions. Okay, which secretes the BMP, maybe 9, BMP9, I think so. BMP9. Okay, these things will be developed. These are the paracrine function, and these two are produced by the Sertoli cells, and a few concentration from the PMC, peritubular myoid cells. These two cases, because of these two, the meiosis will be started after puberty. That means conversion of retinoic acid into gene STRA8 is not inhibiting by the inhibitor protein because lack of it, and the function of the STRA8, that means initiation of meiosis, is not inhibiting by the nanos because it is not present there after puberty. And on other hand, GDNF, that means glial-derived neurotrophic factor and stem cell factor, these are the two factors that are secreted by the Sertoli cells, and PMC, that means peritubular myoid cells, which have peculiar functions. SCF will cause the transition of spermatogonia into primary spermatocytes, and GDNF has the capability to cause the division of type A into another type A. That means cause the mitotic division in the spermatogonial stem cells. Okay, that means increase in the number and conversion into type A single to type A paired, type A paired to type A aligned, where after having a series 4 to 6 mitotic division, this conversion into type A into type B will be caused by this SCF. And if you talk about the BMP9, it has peculiar function. That means there will be receptors for SCF, development of receptors for SCF on spermatogonia is caused due to this paracrine function. When this BMP9 is this concentration is increased, then automatically on the surface of the outer surface of the spermatogonia, the receptors of SCF, stem cell factor, will develop, and those cause the whenever there is a SCF in the cytoplasm, it will come and bind to the receptors and cause the initiation of the signal. That means signal transduction will be caused. Okay, because of this BMP9. Okay, this is the major difference. Why? What happening? And what is the cause behind the timing of meiosis differentiation difference in the ovary and the testes? Now let us see the diagram. Here. Okay. See, whatever I said right now will be here. Ovary. See, oogonia. This is the oogonia. It is mesonephron. That means here the kidneys, from where the retinoic acid will be secreted and reaches the oogonia, and where no CYP, sorry, it is not CRY, it is CYP. Okay, CYP26B1 protein. Lack of this inhibitor protein, the retinoic acid is converted, cause the increase in the concentration of STRA8 gene. Because of no inhibitory proteins in the ovary, the STRA8 cause the initiation of the meiosis. Of course, after some period of time, the meiosis will be arrested at meiosis 1, prophase 1, diplotin 1. Okay, keep it in. Okay. Next, if you talk about mechanism in the testes, retinoic acid will be secreted, and these are the testicular lobules in which the seminiferous tubules will be present. As I mentioned, till 13.5 days, there will be CYP 26B1 will be present and cause the restriction of conversion of production of STRA8 gene by the action of retinoic acid. Thus, retinoic acid will not be cause the initiation of STRA8 gene concentration, which will cause the meiosis arresting. Okay. Next, postnatal. See how this retinoic acid will be formed. Okay. Now it will be discussed here. First, it is the retinol. Okay, alcohol. Here the functional group is alcohol. Whatever the retinol that are present in the cytoplasm of the seminiferous tubules, those will be incorporated into the Sertoli cells. That means it is the cytoplasm. It is the cytoplasm from where this rol, that means retinol is present. It will be entered into the Sertoli cells because of one enzyme, alcohol dehydrogenase. Okay, these convert the retinol into retinaldehyde. See, here the functional group, if you talk about the functional group, it is OH. Here we talk about the functional group, here it is CHO functional group. Now this retinaldehyde will also be converted into retinoic acid, where functional group is COOH, retinoic acid, by the action of retinaldehyde dehydrogenase. Here the alcohol dehydrogenase, here is the retinaldehyde dehydrogenase. These are the enzymes that involve in the conversion of retinol into retinoic acid. Okay, this retinoic acid with the use of the RAR or otherwise, directly it enter into the spermatogonia. This is the Sertoli cells where this retinoic acid is formed. Keep it in mind, retinoic acid formation is happened in Sertoli cells. And by the action of this retinoic acid, the type A spermatogonia cause the transition. And one more thing, initiation of meiosis. Of course, in transition, because of this meiosis only, the transition will be happened. Okay, spermatogonia will be type A, will be converted into type A1, nothing but type As, AB, AS, like so on. Okay, this is the pathway from where retinol is converted into retinoic acid, and those retinoic acid will be entered into the type A spermatogonia, and after conversion of type A, from where it is the serum. Okay, type A into here the expression of the STRA8 gene. Keep it in mind, STRA8. Because of CYP26B1, this STRA8 will be inhibited and converted into so and so. Okay, 4-oxo retinoic acid, all this. Thus, meiosis will be restricted. Okay, next. Here in testis, regularly if we see, it is taken from a another research article. Embryonic testis postnatal. After that, after puberty condition will be also available. See here, RA is present. CYB26B1 is present. It is the gonocyte. When these two come and bind to each other, then degradation of retinoic acid happens and meiosis is here. Conversion is stopping. So because of no striate is not there. Because of that, the meiosis occurrence is stopped. Okay. After postnatal, if you see postnatal, the retinol, already I discussed how the conversion of retinol into retinoic acid. The retinol is present in serum or cytoplasm. Will be entered into the Sertoli cells. It is the Sertoli cell. And conversion of retinol into the retinoic acid happened in the Sertoli cells. Why only there happening, not in the serum? Because serum lacks the ARH and RALDH. That means alcohol dehydrogenase will not be there, and retinaldehyde dehydrogenase will not be there in serum. Those two enzymes will be present, and what are the conditions that are favored for this conversion will be developed or will be given to those enzymes in Sertoli cell. Because of that, retinol is converted into retinoic acid. Okay. Now the retinoic acid will be released into the serum or cytoplasm, which binds with RAR or RXR and cause the entrance into the spermatogonia. Okay. Now spermatogonia, where nanos S is present because it is a postnatal before puberty. Nanos 8 will be present, and also that there will be CYP26B1, which causes the degradation of RA and stops the appearance of the meiosis. After condition already I discussed. So these are the basic pathways and basic mechanisms involved in the meiosis arresting not arresting, inhibition, totally inhibition until puberty in testes. If we talk about ovaries, their inhibition. The word, if we talk about the female gonads, there we can use the name arrest, meiosis arrest. If we talk about the male gonads, then meiosis inhibition. Okay, until puberty. These are the basic differences in the mechanism of meiosis. Now, see, just now I talked about seminiferous people. There will be seminiferous epithelium will be present, Sertoli cells will be present, and spermatogonia, spermatocytes, and spermatids will be present. That means spermatogonial cells will be present. Especially spermatogonia need to be converted into spermatocytes, and spermatocytes will be converted into spermatids. Why only the retinoic acid causing the conversion or initiation of meiosis in the spermatogonia? Why not in the Sertoli cells? Because retinoic acid, there will be constant production of CYP26B1 in the Sertoli cells. When we talk about in spermatogonia, there will be no. Okay, because of this constant production, the retinoic acid conversion in Sertoli cells will not be there. So the retinoic acid will not show its mechanism for conversion of entering a cell into the meiosis when we talk about the Sertoli cells. Okay, that means retinoic acid will not show its effect on Sertoli cells. Only shows its function on spermatogonia. Why? Because only on spermatogonia, because of BMP9 receptors will be developed. Okay, these are the reasons why there is no meiosis occurrence in the Sertoli cells. Okay, next, spermatogonia. What is spermatogonia, and what are the characteristics and the location? All those things will be discussed in this slide. Now see, if you talk about the location, basal compartment. Okay, what is meant by basal compartment and what is the central compartment will be discussed in further slides. It is in the basal compartment. It is the PMC. Above that there will be basement membrane. Adhering to that basement membrane, there will be spermatogonia. Okay, adhering to those basement membrane, there will be spermatogonia. Okay, next, functions. Precursors of sperm initiation, the process of spermatogenesis will happen in these cells. That means spermatogonia acts as the precursor of the spermatocytes, and which will be converted into spermatid, and from where the sperms, after having morphological changes, will be developed. All those things will be happen because of the spermatogonia. Okay, cell type. It is diploid in stem cells, contain 2 sets of chromosomes. That means 23 pairs of chromosomes will be present. Both mitotic and meiotic divisions will be happened in the spermatogonia. Okay, next, if you talk about the control, in under which control means that the proliferation and differentiation. Proliferation and differentiation of spermatogonia are regulated by the hormones and local growth factors, intracellular paracrine. For example, if you talk about the paracrine BMP9, just now I discussed about it, which was the development of receptors on these particular cells. Okay, Next types. If you talk about the types of spermatogonia, there will be two types: undifferentiated and differentiated. Undifferentiated means there will be A single, A paired, A aligned. And if you talk about the differentiated, then it will be B type of cells. Here, C. Okay. Now, what is AS, APR, AL? That means A single, A paired, A aligned. Let us discuss that. See, if you talk about the stages and that involved in the conversion of AS to AP, A aligned, all those things will be discussed later. Okay. See, before that, I will unload it. Keep it in mind. Here is the process. First, there will be AS. That means only single cell will be there. In some books, it is called as the pre-spermatogonium or otherwise spermatogonial stem cells. SMC or pre-spermatogonium. It has the capability of self-renewal. That means production of similar type of cells. And this capability will not be cause any genomic difference. Okay, keep that in mind. That means one cell produces two cells. Out of these two cells, one cell will be restricted for action of stem cell. Okay, another will be entered into mitotic amplification. It is also AS. It is also AS. Okay, one will be converted into stem cell. That means it will act as a future stem cells. Okay, and another will be participated in mitotic amplification. What will be mitotic amplification? That means production of similar types of cells, but with variation in the cytoplasm or the morphological characteristics. Okay, now how many cycles? 4 to 6 cycles. That means 4 to 6 mitotic divisions will happen. Because of this, AS will be converted into AP. That means this cell will be converted, whatever the cell present here, will be converted into two cells. And AP will be converted into A aligned. Because it is the first mitotic division, it is third, like similar, 4 cells will be third, 3, 4, like up to 6. In some books it is recorded as 4, in some books it is recorded as 6. That means in between 4 to 6 mitotic divisions will be happened in mitotic application. That means one cell is converted into 2, 2 is converted into 3, 4, after further, and from 4 to 8, like similarly, production of many cells will be happened. Okay, how many? 2 power 6. If it will be 6 cycles, then 2 power 6 will be the answer. And if it will be the 4, 2 power 4 will be the answer. Those cells will be connected to each other. And those connections are called as cytoplasmic bridges. Okay. After completion of final sixth mitotic division, this A type of cells will be confined to A type B cells. Okay. That means spermatogonia type A, after completion of 6 meiotic divisions or 4 to 6 meiotic divisions, it will be confined to conversion of A type to B type cell. Now, these B cells are the precursors of primary spermatocytes. Okay, these all the mechanism will happen in basal compartment of seminiferous tubule. Okay, keep it in mind. Now, I will show the diagram. Before that, once you have a read. See, spermatogenesis starts at puberty when a pre-spermatogonium undergoes mitotic cell division to produce the two daughter cells. One daughter cell will remain as the stem cell, and other initiate the mitotic amplification, which gives rise to morphologically different spermatogonium type A and type B. Type B completes the. See, very important thing. What will be type B? Okay, type B generally first it follows the mitotic division and enters. We know that mitotic division means there will be two phases: interphase and mitotic phase. In interphase, 3 stages will be there: G1, S phase, G2 phase. Up to now, I thought that every cell which performing the mitotic cell, it will be converted into 2. But here is the one of the different thing I noticed is type B, after performing the G1 and S phase, it will not enter into G2 phase. It will enter directly into meiosis. Okay, it will enter directly into meiosis. See, those things is mentioned. Type B spermatogonium complete the S phase, that means double the DNA synthesis of their cell cycle and advance to G2 instead of undergoing mitotic cell division. Okay, that means instead of mitotic cell phase, they translocate to the adluminal compartment and initiate meiosis 1. That means up to now, whatever the thing happened in the basal compartment. Basal compartment. Up to now, all these things happened in basal compartment, but the after translocation to adluminal, the mitotic phase is arrested. Mitotic phase is arrested, and instead of happening of mitotic phase, directly it enters into the meiosis 1. That is the very important. Okay. Next, a characteristic feature of spermatogenesis is incomplete cytokinesis. Because of these incomplete, the cells are conjoined. Cytokinesis will not be, see, mitotic phase has karyokinesis and cytokinesis. Both karyokinesis is all not happened and cytokinesis is also not happened. Incomplete cytokinesis. Okay, because of these, there will be development of cytoplasmic bridges in between these cells. That means like this, these cells will be. Okay. Until the completion of the spermatogenesis, these cytoplasmic bridges will be present. Another typical aspect of spermatogenesis is cells cycle synchrone. All the cells initiate at a time and complete their cycle at a time. See, all the cells initiate, progress, complete a differentiation sequence in a coordinated manner. The conjoined condition terminates when mature spermatids are released. Nothing but after completion of conversion of spermatids into sperms, then they will be separated into residual bodies and the mature, not spermatids, it is matured sperms. Okay, keep it in mind. These residual bodies will be engulfed by the Sertoli cells, and these are released into the lumen. Okay, those will. This is the mechanism that happen in the spermatogenesis. Okay, now let us see the diagram. This is the diagram. See, pre-spermatogonium A single type is present. It happens self-renewable is seen, and stem cell one will be acted as stem cell, and another will be acted as the cell for the further mitotic amplification, and these enter into mitotic amplification, which causes 4 to 6 mitotic cycles. After completion of 4 to 6, in between 4 to 6, the type A will be converted into type B. Okay. Next, this type B will be entered into mitotic cell division, and cause the interface. Interface will be completed, and after interface, they shifted from basal compartment. All these things happen in basal compartment. From here to these, all things will be happening in abdominal or central compartment of seminiferous tube. Okay. Now, after completion of interface of mitotic cell division in type B cell, they will not enter into the karyokinesis and cytokinesis. Directly, they will shift from the basal compartment to abdominal compartment, and there the initiation of meiosis 1 will be caused, and thus the type B act as the precursor of primary spermatocytes, and because of meiosis 1, these primary spermatocytes will be present, and secondary spermatocytes will be produced. Next, this secondary spermatocytes is converted into round spermatids, and these round spermatids is converted into elongated spermatids with the residual bodies, and these elongated spermatids is converted into single matured sperms plus residual bodies. Okay, this is the mechanism involved in the conversion of spermatogonia to sperms. Next, key feature of this conversion is this is the two. Cytokinesis is incomplete. Factors decides the transition. Those factors is nothing but GDNF, SCF. These are the two factors that produce produced by the Sertoli cells and PMC, peritubular myoid cells. Those two. Next, up to now we talk about the germ cells. Now let us discuss about the somatic cells of spermatic seminiferous epithelium. Somatic cells of seminiferous epithelium is nothing but Sertoli cells. Okay, these cells are also called as substanticular cells or nourishing cells because these cells nourishes the spermatogonia. Whatever may be the structure, it may up to spermatozoon, they will be nourished. Okay. Next, production of the hormones will be seen. What are those hormones? First one, inhibin will be produced, which causes the inhibition of spermatogenesis by controlling the concentration of SFH. What is the mechanism? Mechanism will be discussed whenever I talk about the factors that control the spermatogenesis. At that time, usually I will talk about this mechanism involved in the inhibition of spermatogenesis due to this hormone. Next, anti-Mullerian hormone or Mullerian inhibiting substance. These are the hormone that plays a major role in the determination of sex, not differentiation of sex during the time of this 7th week of gestation, and it produces the androgen binding protein into lumen. Okay. Next, it secretes substances initiating the meiosis, nothing but factors. Those factors are GD, NF, and SCF. Okay. Next, if you talk about the anatomy of seminiferous tubule, based on these cells, 2 parts. Seminiferous tubules is converted into 2 parts. See, I will give diagrammatic representation. See, this is the seminiferous tubule. Let us assume there will be Sertoli cells like this. These 2 Sertoli cells, one Sertoli cell with presence of tight junctions, they are tightly adhered to the other cell. These are the tight junctions. Whatever the structures present outer to this tight junction, it is the near to the basement membrane. Near to the basement membrane. From tight junction to, see if it is basement membrane, it is the Sertoli cell, it is another Sertoli cell. Here is the tight junction, and the area present in between basement membrane to tight junction, it is the basal compartment of seminiferous tube. And from tight junction to inside, along with lumen, totally called as central compartment of seminiferous tubules. In some books, they refer as the adluminal or adluminal compartment. See, those two are mentioned. I will show you clearly. See, these are the region. It is the basal compartment below the tight junction. Okay. About to it. These are all. This region is the adluminal or central. Okay. That means based on this tight junction, the seminiferous tubule is divided into two compartments. One will be basal compartment between the basement to tight junction, and another will be central compartment from tight junction to central plus lumen. Central compartment is also called as adluminal compartment. Now, BTB, that means blood testis barrier. Just now I said that it is the seminiferous tubule. It is the tight junctions. The region between tight junction to basement membrane, it is the basal compartment, and tight junction to inside up to lumen, it will be central compartment. Okay. The tight junctions present from Sertoli cell to another Sertoli cell collectively forms a barrier. That barrier is known as blood testis barrier. See here, the ring of the interconnected Sertoli cells form the Sertoli cells barrier, nothing but blood test barrier. What is the significance of this blood test barrier? See, it causes the prevention of movement of many chemicals from blood into the lumen. So there will be blood vessels outside. These blood vessels cause the blood. Blood contain many chemicals like blood clotting factors and proteins, and all those things will be present. Those are not supposed to be entered into the lumen. Okay. If they entered into the lumen, then they will cause effect to the spermatogenesis. Because of that, they ensure the proper condition for GC, that means germ cell development and differentiation of germ cell in Sertoli tubules. Okay, these two will be provided by this blood testis barrier. If something damage happened to this blood testis barrier, then it caused the abnormality, that is nothing but autoimmune infertility. Now, what are the components of blood testis barrier? There will be cladins, cadherins, germs, that means other molecules will be present, like zona occludens. Of course, zona occludens is 5 types, but only 1, 2, 3 type only will be seen in a blood test barrier. These are the components that makes the tight junctions, and those tight junctions cause the ring-like structure, and those after interconnecting, this seen with, those collectively will be seen as a ring, and this ring is known as nothing but a BTP. That means blood testis barrier. Next, see here you can see that. See, it is the myoid cells. It is the intertubular space. Okay, it is the blood vessel, and these are the lytic cells. These are the lytic cells. It is the myoid cells from where the structure of the seminiferous tubule starts. Structure of seminiferous tubule. See, it is the BTB, that means blood testis barrier. It is formed by the tight junctions. These are the tight junctions, TJ. Okay, BTB. From there to near to the BM, it will be basal compartment, BSC. And after that, BTB to lumen side, it will be the ADC, adluminal compartment. Okay. These are the spermatogonia, A type, B type, and these will be the structures. Primary spermatocyte, secondary, round spermatids, elongate spermatids, all these will be in adluminal. Okay. Next. Now, see, factors secreted by the Sertoli cells plus PMC. A small little amount of factors will be secreted by the PMC, not a major amount. Okay. See, these Sertoli cells produce the two types of factors. One is the GDNF, glial cell lines derived neurotrophic factor. Okay. And second one is the stem cell factor. What is the function of these two? Glial cell lines derived neurotrophic factor determine the dividing spermatogonia last as the spermatogonia, or enter the meiotic phase to become the spermatocytes. It will be determined it will determine the spermatogonia. That means A single type, whether it needs to be converted into A S type, which follow the mitotic amplification, or otherwise it need to be restrict itself towards the A S type of stem cell. It will be decided by this CDNF. Okay, based on the concentration. If it will be in low level, promotes the transition. That means it will be converted into mitotic amplification. It will be leads to cause of the differentiation. That means mitotic amplification. If the high level of GDNF will be present, then self-renewal of stem cells will be seen. It is seen in high level. It is seen in low level. Okay. Next, SCF promotes the transition of spermatogenesis. That means conversion of AS into AB, AB into A aligned, then B, where B is converted into primary spermatocyte. All these things is due to the SCF. And these two factors cause the transition of spermatogonia into spermatocytes. Transition of spermatogonia into spermatocytes. Now, under which control these two will be present? Upregulated by the FSH, follicle stimulating hormone cause this stimulation of Sertoli cells, and these cells produce these two factors. And these two factors serve as the link between Sertoli cells and the endocrine system. Okay, keep it in mind. Next, spermatogenesis and terminology. If I talk about spermatogenesis and the terminology, see, definition: production and release of sperms from the spermatic mother cell. Sperm mother cell. Okay, it is a steady process. That means continuous process. Occur for every 90 minutes. Okay, begins at puberty because significant increase in the secretion of gonadotrophin raising up. Next, per day, how much? 30 million sperms are manufactured. Okay, for completion of one spermatogenesis, around it takes 64 days. Okay, next, total number of mitotic divisions. There will be 2 mitotic division and 1 meiotic division happens. If meiosis, mitotic division, if you talk about the meiotic division, meiosis 1 will be there, and following it, meiosis 2 will be there. Meiosis 1 will be happen in primary spermatocyte. Meiosis 2 will be happen in secondary spermatocyte. If one sperm mother cell follows spermatogenesis, it produces infinity sperms will be produced. From one primary spermatocyte, 4 sperms will be present, 2 X type and 2 Y type will be present. From one secondary spermatocyte, 2 sperms, either of the type, either it may be 2 belong to X type or 2 belong to Y type. Okay. Next, the terminology. What are the terminology and the process involved? Spermatogenesis, the production and release of sperms from SMC. It is under the control of FSH and LH. Next, spermiogenesis, spermatids differentiation into sperm. Okay. Next, semination, sperms released into seminiferous tubules. After the spermiogenesis, semination will be seen. Next, if these sperms that are present in the lumen of the seminiferous tubule is released out from the seminiferous tubules into epididymis through reedy testis, vasa differentia, all those things, then it is called as spermiation. Okay. Spermatogenesis, spermiogenesis, semination, spermiation. These are the technical terms that we need to be thorough. Next, mechanism or process involved. See, mammalian spermatogenesis is highly productive and coordinated process. It mainly consists of 3 phases: proliferative phase, and a meiotic phase, and a differentiation, or nothing but it is also called as spermiogenesis. MIO, spermiogenesis. Okay. First one is proliferative phase. Second one is meiotic phase, where meiosis will be seen. And the third one is differentiation or spermiogenesis phase. Okay. In proliferative phase, and spermatogonia undergoes continuous division and produces the B type of cells. Whereas in meiotic phase, it causes the recombination and produces the primary spermatocyte, secondary spermatocyte, and spermatid. Now, in differentiation phase, the spermatids that are formed from the above phase will be transformed into spermatogonia by happening morphological changes. Okay. Next, the productivity of the spermatogenesis primarily depends on the proliferation phase. Okay. Next, if you talk about the spermatogenesis, spermatogenesis play a pivotal role in the continuity of the male germ cell by producing the haploid spermatozoa that fertilizes the egg and eventually produce progeny for the next generation. It is the significance. Okay. Next, spermatogenesis chart. See, it is the chart prepared by me. It is very, very important one. Without understanding the previous slides, you can't go directly into the spermatogenesis chart, because with having the proper knowledge and the difference and whatever the structure of the testis and seminiferous tubules, then only you can understand this step. See, there will be PGC first. Okay. For every cell, there will be ploidy, number of chromatids, number of chromosomes, and where the meiosis occurred, mitosis occurred, all those things will be included here. See, first, part of this seminiferous tubules, name of this cell, number of chromosomes per cell, will be given here, and number of chromatids will be present. Number of that means ploidy. What the ploidy in the particular type of cell, and division. Either it is meiotic division, either it is mitotic division. If it is meiotic division, then which type of meiotic division, meiotic 1 or meiotic 2, will be discussed here. Okay. See, let us discuss that. First, there will be PGC. PGC will be converted into SMC. Okay. Now, spermatogonia or spermatogonial stem cells will be present. These all things happen in basal compartment. PGC will be migrated into gonads, and in gonads, seminiferous tubule is converted into basal compartment and central compartment due to the presence of it is BTB or tight junction. Okay, tight junction. Now, here to basement membrane. It is the basal compartment. In basal compartment, spermatogonial stem cells, which has the 46 or 44 plus XY. Number of chromatids will be 2. Ploidy will be 2N or diploid ploidy. Division. If you talk about the cell, SMC, undifferentiated and unmatured cell, when there is mitotic amplification, it causes the development of type B and type A cell. Type A will be ruled out of the spermatogenesis and act as the stem cell for future and follows mitosis. Type B, primary spermatocyte. See, type B will act as the precursor for primary spermatocyte. What will happen? B will follow mitotic cell division and completes the interphase. Interphase will be completed, but will not be occur mitotic phase. That means karyokinesis and cytokinesis will not be seen. Directly, without happening this, it will pass from basal compartment to adluminal compartment or central compartment. Okay. Now, what the difference between the primary spermatocyte present in the basal compartment and central compartment? Only difference by size, volume, and the immunological functions that is present in those two cells. The one present in the primary spermatocyte, it will be little bit small, and the one present will be here will be more. Okay. Next, primary spermatocyte. If you talk about the number of chromosomes, it will be like this, and the number of chromatids will be same as these cells, and ploidy also same, because only mitotic division, no meiotic division is happened until now. Okay. Now, in primary spermatocyte, the meiotic 1 will happen. See, if you talk in primary spermatocyte, meiotic division will be caused. It is under the stimulus of retinoic acid, a derivative of vitamin A. Okay. When 24 days. Okay. Now, this primary spermatocyte is converted into secondary spermatocytes. By the occurrence of meiotic division 1, the primary spermatocyte produces two secondary spermatocytes, set A 22 plus A and set B 22 plus B. That means in meiotic division 1, the homologous chromosomes will be separated. That means ploidy will be reduced. Because of that, the ploidy 2N converted into N, and 46 is converted into number of chromosomes per cell, converted into 23. 44 plus X Y is converted into 22 plus X or 22 plus Y. This will be the number of chromosomes per cell, type of chromosomes. Okay. So these are the secondary spermatocytes. Now, following it, here is the another one. Now, secondary spermatocyte. It is 22 plus X type, it is 22 plus Y type. We know that meiosis 1, whenever we are talking about meiosis 1 will be reduction division, and meiosis 2 will be equal division, similar to mitotic division. So the ploidy will not be deferred. Secondary spermatid 22 plus X within the 8 hours completes the meiotic division 2 and converts into spermatid. So similar type of cells will be formed. 22 plus X only will give. If it is 22 plus Y type of secondary spermatocyte, then it will produce 22 plus Y type of spermatid. Okay. Now, from which spermatid is converted into sperm? Here the follows the third step. Nothing but spermiogenesis. That means conversion of spermatid into sperm or spermatozoa. See, these are all things happen in central compartment. Okay. These are the sperms. Finally, 4 sperms will be produced. Okay. And those will be released into lumen of seminiferous tubule. Okay. Next, methods. For separating of X and Y bearing sperm, we follow the flow cytometric or flow sorting techniques. These are the two techniques through which we can separate the sperm which contain the X. That means if we need to separate the 22 plus X type of sperm from 100 sperms, then we follow the flow cytometry or flow sorting techniques. Okay. Next, what will be the type of questions that will be asked? See, the questions may be spermatogonia rested in which part of the seminiferous tubule? One type of question. Spermatogonia consists of which ploidy, or number of chromosomes, or number of chromatid per chromosome, or spermatogonia will be developed into which type of cells? Okay, these type of questions will be asked. And meiotic division will be caused to the production of which type of cells, or meiotic 1 will cause in which type of cells, These are the questions that will be arising from this topic. So if you are thorough with this chart, then surely you can answer those things. Next. Okay. Why I place this slide and marked only the primary spermatocyte? Already I mentioned that. See, there is two terms we need to concentrate. One is N and one is C. See, if you are talking about the N, number of chromosomes is notating. If you're talking about the C, the amount of DNA is to be quoted. That means number of chromosomes is totally different when we compare with amount of DNA. That is the peculiar thing you need to be noticed here. If you notice that thing, then there is one of the important thing that you can learn from this slide. Okay. See, in spermatogonia, diploid 46 will be present. If you talk about the number of amount of DNA, 2C will be present, and number of chromosomes 46 will be present. That means 2N. Okay. Now, see, let us talk about primary spermatocyte. Diploid 2N, 46 is present. Here 4C. Why it became 2C into 4C? Because, as I already mentioned, spermatogonia is converted into primary spermatocyte type B, especially type B, following interphase, but not cytokinesis or karyokinesis. That's why there is presence of 4C. That means doubling of DNA is happened. After that, there is no karyokinesis or cytokinesis. Thus, 2C is not converted into 2C, 2C. This 2C is rested like that only and converted into 4C. Because of that, the primary spermatocyte has the 4C. That means double amount of DNA. It is the most important one to be noticed. Okay. See, if you talk about the meiosis 1 and meiosis 2, meiosis 1 is the separation of homologous chromosome where the N will be divided. That means if it is having 2N or 2N, it will be converted into N. Okay. And this N, after completion of meiotic division 2, then N will be rested like that. Okay. Simple thing. Only meiosis 1 cause the separation of homologous chromosome. Meiosis 1, meiosis 2 occur in primary spermatocyte. Meiosis 2 occur in secondary spermatocyte. Cause separation of homologous chromosomes. Separation of chromatids, sister chromatids. Next. Ploidy. 2C is converted into 2N is converted into N, whereas here N is rested like that only. No reduction. It is called as reduction step of meiotic division. It is the equational division. See, equational division. Division of meiotic division. This is the major difference. That's it. Okay. Next. See, this is the overall procedure and the structures and different types of phases in the cells. See, first PGC will be present. After migration, gonocytes will be reach the gonads and called as gonocytes, where gonocytes is developed into type A spermatogonia. After puberty, cycling of stem cells, ploid will be 2N, and intermediate spermatogonia, that means A, it is the AS type. AP will be developed, A aligned will be developed, aligned in terms of 4 cells, in terms of 8 cells, in terms of 16, in terms of 32, and committed spermatogonial cells next will be final in mitotic amplification. Final cells. Those cells is converted into type B spermatogonia. That these act as the spermatocyte precursor cells, in which mitotic division, the two only interface is same, and cause the movement of this type B into adluminal. There, the primary spermatocyte will be seen. It undergoes the meiotic division 1, where the recombination is seen, and thus the 2N is converted into N and produce 2 secondary spermatocytes, in which meiotic division 2 is seen. Okay, which is similar to mitotic division. So there is no difference in the ploid. Okay, are produced around the spermatid. These undergoes spermiogenesis stage and converted into elongated spermatid plus residual body. These are phagocyted by the Sertoli cells. These residual bodies are phagocyted by the Sertoli cells, and these elongated spermatids will be formed into matured spermatozoa or sperms. Okay, this is the overall process that is present in the spermatogenesis. It is the chart. Okay, next. Spermiogenesis. Spermiogenesis, not spermatogenesis, spermiogenesis. Sperm metamorphosis. Okay, spermogenesis is also called sperm metamorphosis. Okay, what will be discussed? One, definition. Two, morphological changes or process involved in differentiation. Okay. Next, third one, structure of sperm and characteristics of spermatids. Okay. These three will be discussed under the spermiogenesis. Okay. Now, first one, definition. What is meant by spermiogenesis? That means differentiation of spermatids into spermatozoa is known as spermiogenesis. Okay. This process takes around 2 to 3 weeks. Spermiogenesis takes around 2 to 3 weeks, and a series of extraordinary events happen, and this structure will be converted into spermatozoa or sperm, which is also called as sperm. Okay. Now, if you talk about the characteristics of spermatids, it will be without flagellae, because of that non-motile in nature. If you talk about the ploidy, it will be haploid in nature, as these are the products of meiosis II, which happen in the secondary spermatocytes. Because of that, it is haploid in nature. And if you talk about the shape, it is round. Okay. If what are the cell organelles that are present means nucleus will be present. Then Golgi apparatus will be present. These are the cisternae of Golgi apparatus. Okay. Mitochondria will also be present. Along with that, centrosome will be present. We know that centrosome is the structure that contains a pair of centrioles. Okay. Centrosome will be present. Now, what will happen in spermiogenesis? See, superfluous material that are present in these spermatids will be discarded, and certain organelles, that means not all organelles, certain organelles are become specialized. Okay. What that specialization? Let us see the specialization. See, spermatid, if you talk about the shape, round, this is the sperm, it is elongated cell. First, what are the structures that are cell organelles present in the spermatid? I will discuss that. Then, which structure or which cell organelle is specialized into which form will be discussed later. First, nucleus will be present, Golgi apparatus, centrosome present, mitochondria, cell membrane. See, there will be cell membrane in both. If you talk about the cell membrane in sperm, it will be present at everywhere except at end tile, except at end tile of tile region. Other than these, remaining all structure will be covered by this plasma membrane or cell membrane. Okay. Next, if you talk about the sperm, the Golgi apparatus present in the spermatid is specialized into acrosome pair. Now, what are the centrosomes that are present in the spermatid is specialized into two centrioles which have peculiar functional properties. That means if you talk about the proximal centriole, then it involves in the formation of spindle fibers and controls the cell division. Like similarly, each centriole have specific function at specific time. Okay, those will be discussed in further slides. Next, if you talk about the mitochondria, here the mitochondria will be in individual form, and whereas if you talk about in sperm, the mitochondria will be specialized into a single sheath. All the mitochondria unite and forms a single sheath around the distal centriole and axial filament. Those sheath is called as Nepekins sheath. All will be discussed in upcoming slides. Okay, these are the specializations that we can see in this cell organelle of spermatid. Okay, now mechanism or procedure involved in differentiation. See, spermatid is a round structure without head, without middle piece, or otherwise tail region, without neck. But while differentiation, these will be formed, and thus sperm with head, neck, tail will be seen. Okay. Now, if you talk about the mechanism, the formation of these three will be seen. Okay. See, first one, formation of head, formation of neck, third one, formation of tail. These are the procedures that involve in the differentiation of spermatozoa. Okay. Now, not spermatozoa, differentiation of spermatid. Let us see. Formation of head. See, head of sperm contains nucleus plus acrosome. Okay. Whatever the changes that happening during the formation of nucleus and during the formation of acrosome, if we discuss about those two events, then those events compose the formation of head. Okay. Now, changes that happening in the nucleus. See, spermatid is present. Here the spermatid is there and the nucleus is there. It is in round shape. Okay. Now, thus this spermatid is differentiated into sperm. It also contains nucleus. Here the shape should be elongated. That means first shape is round and spherical is converted into elongated and narrow. Why this conversion? That means one form to another form of shape is why. Because two functions. One, propulsion in any fluid will be increased and penetration of this sperm into the ovum will be enhanced because of these reasons. For example, how? If you talk about the propulsion and penetrancy, capacity or affinity, it basically depends on surface area. If the surface area is more, then the repulsion exerting towards it will be very high. It will be very high, not less. It will be very high. Surface area is more, then repulsion also will be more. Okay. Now see, if you talk about the penetration capacity, here spermatid is present, and on other hand, the female gamete ovum is present. See, when these two, here the nucleus is present. When these two comes in contact, then if we see the surface area, there is more surface area. So there will be more repulsion. Thus, it will take a lot of time and the penetrancy capacity will be very low. Now, if it is elongated, see, it is elongated like this, then the point of surface area that is coming in contact with the female gamete will be very less, very less when compared with the round shape. Thus, the repulsions will be very less. Because of that, easy penetrancy is sent. Penetrancy is notation. Because of that, the penetrancy capacity will increase. If we talk about the propulsion, see, round nucleus is present. If we kept it in a fluid medium, so now the area of contact to the fluid will be more. Because of that, the repulsion exerted by the fluid on the structure will be more. Thus, forward movement will cause slower. When it is in elongated form, the contact area that is coming towards the fluid medium will be very less, and the other fluid exert on the other form in not directly on 90 degree. It will exert less than or more than 90 degree. Because of that, when we compared, there will be very less repulsion is noticed. Thus, it can move forward in any medium more faster. Okay, this is the propulsion or penetration affinity towards the related to shape. Okay, next size. If you talk about the size, nucleus basically consists of nucleoplasm and chromatin substances. See, most of the nucleoplasm is composed of water, and those water is out, lost out from the nuclear cap. Okay. Next, if you talk about the chromosomes are closely packed, thus the volume will be decreased. Because of these two reasons, the size of the nucleus will reduce very significant. Okay. Next, if you talk about the nuclear membrane, the pore complexes that are present, we know that nuclear membrane is double membrane with pores. Those pores are filled with pore complexes. These pore complexes are lost during these events. That means during the changes that is happening in the nucleus differentiation. At the time, these pore complexes that are present in the nuclear membrane will be lost. Okay. Next, histone proteins. Very important. Histone proteins. Now see, I will take a new slide. histone proteins is there, which are rich in lysine plus arginine. Okay, lysine plus arginine. Next, initially these histone proteins, basic function of these histone proteins is chromosomal organization, because these are basic amino acids with the positive charge. Okay. Now, during this differentiation of nucleus, the histone proteins is initially replaced by transitional proteins, which plays a major role in transportation or trafficking, cellular trafficking. Later on, these transitional proteins are finally or later on replaced by protamines. Okay, protamines. Now keep it in mind, protamines are the structures which are small proteins rich in site cysteine plus arginine. Okay, what the significance of this protamine? Higher compact, high degree of compaction or higher compaction of nuclear chromatin. It is the function of protamines. Now keep it in mind, it is the replacement one. It is the another replacement two. Okay, replacement one, that means replacement of histone proteins by transitional proteins happens at the time of changes that is happening in nucleus. That means differentiation that is happening during the time of change in nucleus, this replacement will be seen. And another replacement, that means transitional proteins are replaced by the protamines. This replacement is seen during the development of acrosome. At the time of development of acrosome, these are the two events that take place during different times that have happening during the time of formation of head. Now keep it in mind one thing. See, these transitional proteins that are present, is replacing by these protamines. These protamines. That means already protamines are formed and present in nucleus. Okay. When these protamines are present are formed, keep it in mind, precursors of protamines formed in primary spermatocytes. These precursors of protamines will not accessible to transitionary mesh until the spermatid stage is formed. Okay, until the spermiogenesis is started, these precursors of protamines will not be accessible to transitionary meshes. Thus, these precursors of protamines will not be converted into proteins. Okay. Whenever this spermiogenesis, that means spermatid is formed and spermiogenesis is starting, at that time these precursors of protamines will be available, will be accessible to these transitional machines, and thus the formation of the protamines is taking place. Okay, precursors of protamines is formed in which stage of the cell means primary spermatocyte, and protamines formation is happened in which stage of the cell means then it is a change that is happening in the nucleus. Okay, keep it in mind. Next. See, here the point is noted. First synthesized in primary spermatocytes but are not translated into proteins until the spermatid stage because inaccessible to transitionary message. Okay. Next, if accessible, chromosomes condense prematurely and sterility results. Okay, if suppose those proteins is accessible to transitionally machines, then it will lead to the chromosomes condensation earlier than this spermiogenesis and results in the sterility. Next, position. Nucleus moves towards one pole of the cell to form the head of the spermatozoa. Okay. Next, if you talk about the weight, drastically it is reduced. Weight of the nucleus is reduced drastically because the elimination of RNA, nucleolus, most proteins that are present in the nucleus is seen, and because of that, the weight is reduced drastically. Okay. This is the changes that is happening in the nucleus. Next, development of acrosome. See, in development of acrosome, Golgi apparatus is the main structure which act as the precursor of the acrosome structure. Okay, that means Golgi apparatus leads to the formation of the acrosome after having some significant events. That means this development of acrosome contains four sequential phases, that is G-CAM as a short notation. G means Golgi phase, C for cap phase, A for acrosomal phase, M for maturation phase. These features. Okay. As we all know, G-CAM. Okay. First phase. G stands for Golgi phase. See, what will happen in Golgi phase? First one, formation of acrosomal vacuole is the first one. Second one, centrioles formation. Third one, axoneme assembly. Initiates or starts. Okay, keep it in mind. These are the key events that happen in the Golgi phase. Let us discuss about each one. Formation of acrosomal vacuole. First, I will discuss about the formation of acrosomal vacuole. See, Golgi apparatus is present. Golgi apparatus produce glycoprotein granules, small glycoprotein granules, which are called as pre-acrosomal granules. Pre-acrosomal granules. Now, these various number, that means if let us there is 10 pre-acrosomal granules. Now, these 10 will unite and form one big vacuole. This vacuole now called as acrosomal granule or vacuole. Okay, thus the process present in the formation of acrosomal vacuole. Okay, next, see, if we see, let us, it is the seminiferous tubule. Here the central cells will be present, and like this there will be in center, it is the lumen, it is the central cell. If you take this part and draw separately, then like this, PMC, BM, upon that there will be central cells. In between there will be spermatogonia based upon site, basal compartment or central compartment. Respective cells will be present. Now we are talking about the spermatid. So spermatid is present here like this, and other two central cells by othering junctions. Othering junctions. Okay. Now see, it is the cell which contain the nucleus. Nucleus will be present. Okay. Now, towards the center, here the acrosomal vacuole will be formed, and here will be the Golgi apparatus. These are the Golgi apparatus. These are the acrosomal granule or vacuole will be present. It is the lumen, near the lumen side. See, next, that means keep it in mind, the acrosomal vacuole will be formed towards the lumen, while the formation, it is present towards the lumen, not towards the central cells. Okay, that is the main thing. Next, second one, centrioles formation. How centrioles will be formed? During the meiotic divisions, already we discussed in the previous slide that cell organelles will be become specialized in this differentiation process. That the centrosome which is present will be divided and it forms into two centrioles. Meiotic divisions leads to formation of two centrioles towards the lumen side. Towards the lumen side. Now, centrioles already formed in the process of meiosis. Now, these centrioles migration will be seen, not formation, migration. Centrioles migration. Migration, not formation. Centriole formation happens due to the meiotic division 1 and 2. That means one chromosome will be differentiated into two centrioles. It's there itself. And now the centrioles that is formed towards the lumen side will migrate. That means centrioles formation happens here. But whenever this Golgi phase is continuing, the centrioles present towards the acrosomal vacuole will be migrated opposite to it. That means, see, here the nucleus, acrosomal vacuole present here. Here the centrioles will be present. Now they slowly shift towards the opposite end of the acrosomal vacuole. Acrosomal vacuole is present this end. It will be shifted to, that means centrioles will be shifted to opposite to that pole. This, like here. Here they will be. They will lie perpendicular to each other. One will be along the axis of this, and another will be horizontal axis to the spermatid. Okay, like this they will be arranged. Keep it in mind. The central migration happened in which phase means Golgi phase. Those centrioles shift towards the opposite pole of acrosomal present pole. Okay. Next, axoneme assembly initiation. Now see what will happen. See, it is the spermatid. Here the nucleus will be. Now acrosomal vacuole formation. Vacuole formed here. Acrosomal vacuole. Now there will be two centrioles. This is one centriole and another will be another centriole. Okay. Wait a minute. Direction also very important. It is the proximal centriole. It is the distal centriole. See, keep it in mind. Proximal centriole is always perpendicular to the axis of spermatid. And DC, that means distal centriole, is parallel to the axis of spermatid or sperm. In both, it will be parallel to the their longitudinal axis. That means now see, from the DC, the axoneme assembly, that means axoneme assembly initiates. These are the three key events that happens in the Golgi phase. Next, C. C stands for cap phase. Whatever the things happened in upper will be either promoted or discarded. That means acrosomal vacuole present in upper one for perform a specific function here. See, if we talk that first one, acrosome vacuole formed here. Now it becomes, that means 1 minute. Acrosomal vacuole become flat and initiates descent over the nucleus. Okay, it is the first key event. Second key event, axoneme, which 1 minute. Spermatid rotation. It is the key event. Spermatid rotation. Very, very important. Spermatid rotation is this another key event. And the third event, axoneme extension into the lumen of seminiferous tube. These three are the key events. See, already whatever discussion in the Golgi phase, those will be promoted or discarded in the in cap phase or acrosomal phase or maturation phase. See what will happen in cap phase. Acrosomal vacuole which are present here, it will become flat. See, I will show you. See, acrosomal vacuole. Like this, it will be present. It is the spermatid. Here the nucleus is present. Actually, nucleus is not round because already we discussed it. Nucleus will be elongated. Shape is elongated. See, here the acrosomal vacuole is present. No? it slowly starts extending like this. See here, the extension is happening. Its initiation towards the whole nucleus will initiate. That means spreading of acrosomal vacuole will be seen in this phase. Next, spermatid rotation. See, already I informed it is the Sertoli cells. 1 minute. It is the Sertoli cells. Towards the Sertoli cells, see here the Sertoli cells will be present. Towards the Sertoli cells, axoneme assembly is initiated and promoting is same. Now see, if it is developed like that, it will be incorporated into the Sertoli cells. So there need to be shifted. The direction need to be shifted. So the spermatid which are present here, it will rotate and becomes like this. Sertoli cells will be present here. Similar, the spermatid present like this. Then nucleus will be like this. See the extension, acrosome extension happening like this. Okay, and here the distal proximal, next distal. Now the already initiation happened in a Golgi phase now. Which one? Axoneme initiation. Now it prolongs and cause the development of, it is lumen side, because this is the BM. So it is lumen side. In lumen side, the axoneme development promoted. That is the main one. Okay. Next, see, A stands for acrosomal phase. What will happen in acrosomal phase? First one, Manchete formation will be seen. Next, second one, mitochondria. Up to now, they are in individual form. Starts merging and arranging around the distal centriole. Next, third one, Golgi apparatus up to now which are present will reduced and discarded as Golgi rest along with cytoplasm. These three are the key events that happen in the acrosomal phase. That means Manchete formation. What will be the composition of Manchete? That means formation. Manchete means microtubule formation. Microtubule assembly is started in this phase. Now mitochondria starts merging and arranging around the distal centriole, and in future, during the formation of the neck, they forms into sheath. Golgi apparatus is discarded as Golgi rest along with the cytoplasm. These three are the key events of acrosomal phase. Next, see, M stands for maturation phase. That means the key events are Manchete, dissemblance, disassembly is seen here. Next, chromatin condensation. Chromatin condensation happens. Next, third one, cytoplasmic bridges. Cytoplasmic bridges broken. Now see, Manchete formation happened in the acrosomal phase will be disassembled in maturation phase, and they will be discarded. And next, second one, chromatin condensation happens. Because of this, the nuclear type chromatin is converted into somatic or simple type, not somatic, simple type. Sorry, not somatic, simple type. Simple or smooth type chromatin. Nuclear type of chromatin condensed into smooth type of chromatin. Next, cytoplasmic bridges. See, up to formation of this completion of maturation phase, there will be cytoplasmic bridges present between spermatids. Okay. Now these cytoplasmic bridges will be broken and leads to the formation of residual bodies plus mature spermatids. Mature spermatids. Now, what will happen? See, these residual bodies will be phagocyted by Sertoli cells. And these matured spermatids will be released into lumen. But keep it in mind, what are that means mature spermatids is not physiologically mature. That means in terms of physical, morphologically, it is matured, but not functionally. When it reaches the epididymis, then it becomes the functionally or physiologically maturation. See, maturation is of two types: physical or morphological. By the end of this maturation phase, that means by the end of this spermiogenesis, they become morphologically matured. Okay. Another type is physiological or functional. Okay. This happen at the time when the mature spermatid reaches the or sperm reaches the epididymis. There, the secretions present in that region cause the functional maturation of this sperm. Okay. This is the phase, phases involved in the development of acrosome. Okay. Now let us go to the slide. There, diagrammatically, we can see that. See, Golgi apparatus is there. It will convert into acrosome. 4 sequential phases will be there. Golgi, cap phase, acrosomal, maturation phase. What will happen in Golgi phase? Golgi apparatus is present. It secretes the glycoprotein-rich granules, referred to as proacrosomal granules. These granules is many in number. Okay, those fuse and form a single large vacuole, acrosomal vacuole or granule attached to the nuclear membrane. That means nuclear will be like this, attaching to this nuclear membrane, there will be formation of acrosomal vacuole. Now, cap phase. Acrosomal granule shifts to the tip of the elongated nucleus. Granule loses its fluid content and spread over the nucleus and form the head cap. Okay, keep it in mind. What will happen? The Golgi apparatus, which secretes the acrosome, separates from the head cap and move towards the opposite pole. Okay. Next one, the centrals which are close to the nucleus on the side opposite to the acrosomal cap develop a flagellum, nothing but axonal development. Next slide. See, acrosomal phase. Head of the developing sperm contained in the acrosome and the condensing nucleus remains embedded in the Sertoli cell because these are the nourishing cells. The growing axoneme extends into the lumen of the tubule, that means seminiferous tube. Golgi apparatus is gradually reduced and ultimately discarded from the sperm as Golgi rest, along with the some cytoplasm. In term, what will happen in maturation phase means that it is the final phase in development of acrosome. Okay. Unneeded cytoplasm is shed as residual body from each spermatozoon and remaining intracellular bridges are lost. Mature but not yet functional sperm. Mature but not functional sperm are released into the lumen of the but not functional. Mature but not functional. Sperm are released into the lumen of the tubule. See, if you see clearly, here the matter will be. If you see the diagram, here the spermatid is present. Here the axoneme initiation happened. See, acrosome vesicle. Here the acrosome vesicle. 1 minute. See, there is dot-like structures. I will enlarge it, then you can see. See, now you can see. See, here the Golgi apparatus is present. They secrete the granules, proacrosomal granules, and they all fuses and forms the single acrosomal vesicle. It is the first key event, and second, migration of centrioles, and third is the axoneme assembly starts. Okay, see, it is the nucleus opposite to the acrosomal. The centrioles migration is happened, and those from the it is the proximal, it is the distal. From distal to there is axoneme assembly. Axoneme assembly starts. Okay. Next, if you talk about the gap phase, see here the rotation. That means here it is present this side, it is rotated. Now see here the rotation is rotation. Now acrosome. Here the central cell will be present. Okay. Now the acrosome is towards the central cell, and here the lumen. Lumen side axoneme is developing. Okay, from the distal centriole. Next, this is the acrosome becoming flat by loss of cytoplasm present in that. And see, this is the place where acroplagosome. See, what is plagosome? See, acrosomal sac flattens, forms a cap attached to the nuclear envelope through the acroplagosome, and F actin containing cytoskeletal plate. It is the cytoskeletal plate. Okay, and initiates its descent along the nucleus. Okay, next, axoneme continues its growth. Already I mentioned it. Okay, it is the thing. Next, if you talk about the next stage, acrosomal phase. See, in acrosomal phase, what will happen? See, acrosome continues and it descends attached to the acroplagosome. See, manchity formation. It is the green color one is then. It is manchity. What is the combination of what is the composition of manchity? Means microtubules plus perinuclear ring, because it is the nucleus. Around it, a ring-like structure will be formed. So outside the nucleus, a ring shaped. So perinuclear ring of manchity is present. These are the microtubules. Okay. See, if we see this diagram, particularly this diagram, axoneme, mitochondria. See, mitochondria arranging is in this phase around the axoneme and the central distal centriole, around the distal centriole and axoneme. Okay. Outer dense fibers is assemble of outer dense fibers happen in this phase. Okay. These are the acrosomal phase. If we talk about the maturation phase, it is the next phase. See, acrosomal phase. After completion of acrosomal maturation phase, see, cytoplasmic bridges is there. This will be removed, and these residual bodies will be separated from the matured spermatid. Here it is mentioned. The manchity dissembles upon completion of the nuclear elongation. See, nuclear elongation completes at this phase. Okay, one of the key elements. The chromatin condenses, that is somatic histones, are replaced by the protamines, and an enclosome type of chromatin changes into smooth type chromatin. That means when there is histones, formation of nucleosomes is seen. That means chromatin organization, chromosome organization is happened. So nucleosomes formation is seen. Now this histone proteins is replaced by the protamines. So the protamines cause the high degree of compaction of a nuclear chromatin. So the nucleosome type chromatin changes into smooth type chromatin. Okay. Now residual bodies linked by cytoplasmic bridges separate from the mature spermatids at spermiation. Okay. Now these residual bodies which are separated will be engulfed by the central cells. Okay. The matured spermatids will be released into lumen of the seminiferous tubules, and this process is known as spermiation. Okay. Next. See, thus formation of the head is completed, and next step in the differentiation of spermiogenesis is formation of the neck of the spermatozoa. Now see, meiosis 2 completed, thus creates two centrioles. Okay. First, what is centrioles? Two cylindrical bodies. Okay. Okay. These centrioles lies perpendicular to each other. That means one will be longitudinal, then other will be surely it is horizontal. It is longitudinal, and other will be this. So the angle between these two will be 90 degrees. Okay. Keep it in mind. Next, during the early stage, two centrioles move to a position just behind the sperm nucleus. See, it is the spermatid. Let us assume it as a spermatid. See, here is the nucleus. Now, here is the central cells. Central cells. Here is the basement membrane. It is the lumen. So, two centrioles is present. Two will be present near the nucleus. Okay. The one present near the nucleus will be developed into proximal centriole. The one present above this towards the lumen is called as distal centriole. Okay. See, two centrioles move to a position just behind the sperm nucleus. A depression is formed in the posterior surface of the nucleus. That means here it is anterior, it is the posterior. Towards the posterior of nucleus, formation of the depression, and in that depression, one of the two nucleus become placed, and its axis approximately right angles to the main axis of the spermatozoon. That means it is the axis of the spermatozoon means then the proximal will be right angle to this. Okay, I will show clearly. See, it is the spermatid. Here will be the nucleus. See, it is the axis of a spermatid. Now, here a small depression will be seen. In that depression, proximal centriole will be placed. Okay, this is the axis of proximal centriole. That means the angle between these two will be 90 degrees. Okay, next, what is the function of the proximal centriole? Cleavage is the main function of the proximal centriole. Okay, it plays major role in the cleavage. Okay, how it play major role? That means by giving rise to the spindle system. Okay, initiation of cell division in fertilized egg is caused by this spindle system. Okay, keep it in mind. Next, just behind the position of the proximal centriole, one with its axis coinciding with the longitudinal axis of the spermatozoon, a centriole will be placed. That centriole is called as distal centriole, which gives rise to the axial filament. Axial filament, not axis filament, axial filament, which serves as basal granule. Thus, gives rise to the axial filament. Okay. Now, if you see clearly, here the nucleus will be, it will be the PC, it will be the DC. That means distal centriole. This side will be the Sertoli cells, from where axonium develops. It is the lumen side. Okay. The structure will be like this. See, it is spermatid, which is undergoing differentiation. Spermatid, which is undergoing differentiation. Okay. PC and DC. See, it is the axis of spermatid, and which is similar to the axis of the DC. Okay. The axis of DC and spermatid is same. That is the point to be noted. Okay. With another point is PC, proximal centriole, is perpendicular to both DC and spermatid. It is the another point. Okay. Thus, the formation of the neck of the spermatozoon is formed. Okay. Next, formation of tail. Now see, keep it in mind, most of the mitochondria of spermatids concentrate around the, already I mentioned, in acrosomal phase, I think acrosomal phase of development of acrosome. Already I mentioned that mitochondria start assembling around the axial filament and DC, distal centriole. See, it is the mitochondria. They form assembling, okay, around the DC and AF. That means axial filament. Okay, proximal part. That too extremely proximal part of axial filament. Now, the see, tail is the mixture of middle piece plus principal piece plus end piece. I will show the picture. 1 minute. See, if you see clearly, it is the tail part, which is composed of middle piece, principal piece, end piece. It is the head region, it is the neck region. Which one? This is the neck region. Okay, keep it in mind. That means tail is the combination of middle piece plus principal piece plus end piece. Okay, now see. The middle piece of the sperm, the mitochondria lost their individuality by fusing to a great or lesser extent. That means whatever the mitochondria present, in the around the DC and proximal part of the AF, they lost their identity, individuality, not identity. They lost their individuality and combined into a large sheath. Those sheath is nothing but Nabenken sheath, Nabenken sheath, which act as the energy source for swimming of sperm. Okay, these mitochondria act as mitochondrial sheath, Nabenken sheath act as the energy source for swimming. Now see how the middle piece and principal piece and end pieces are formed. See annulus. See before that, what is annulus? See already I mentioned that there will be two centrioles. Okay, in the neck region and the initial position of middle piece. Initial position of middle piece. Okay. Now see another a dark ring is present, which is called as ring centriole at the posterior end of the middle piece. See, it is the middle piece. It is the middle piece. Here the DC will be present. Here the, if it is the structure of sperm, it is the head region, it is the neck region. See, in the starting of the neck region, it is the PC, and ending it is the DC. Just at the start point of the MP, the DC will be present. At the posterior of MP, there will be one more, which is ring centriole. Okay, it is the connection between the middle piece and the principal piece. Okay, it is also called as annulus or Jensen's ring. Annulus or Jensen's. Now, middle piece is formed by the annulus plus mitochondrial sheath plus some part of axial filament. Okay, these three combine and form the middle piece. This middle piece is surrounded by plasma membrane, completely surrounded by plasma membrane. No exception is present in middle piece with related to the plasma membrane. Now, remaining part, whatever the remaining part left in the axial filament, that elongates and forms principal and end piece. See, principal piece also completely surrounded by plasma membrane, but the end piece without plasma membrane is seen. Question: which part of tail lacks cell membrane or plasma membrane or leaving membrane? Answer is end piece. Okay, keep it in mind. Tail of sperm. Okay, now see, if you see this picture, you will get clear over the. See, it is the PC. It is the nucleus. That means it is the head region. Up to here, the head region is present. From here to here, the neck region will be present. See, here is the PC. It here the DC started, and DC will be like this, and because it gives us the axial filament, so it is axial filament. See, it is the axial filament. See, the outer one, the blue color, sky blue color. It is the outer dense fiber. Outer dense fibers. See, outer dense fibers associated with the outer microtubule doublets of the axon. Okay, if you see clearly, if you want to see clearly, I will unload it. See here, it is the nucleus part, it is the head, it is the neck, it is middle piece. See, whatever the white colored bandage type rings, it is the mitochondria. See here, the mitochondria, it is the mitochondria. Okay, mitochondrial sheath, not mitochondria, mitochondrial sheath. Inside that there is axoneme. Okay, axoneme, if is just in between the mitochondrial sheath and axoneme, the outer dense fibers is present. ODF is present. Okay, ODF has transverse ribs. Keep it in mind. Okay, it is the longitudinal column will be present. Okay, it is the structures. Next, thus first formation of head is discussed. After that, formation of neck is also completed. Now the formation of tail is also completed. These three happened in the spermiogenesis. spermiogenesis. Okay. Now, what is the structure of the sperm after completion of spermiogenesis? That means the conversion of the spermatid into sperm. What will be the structure and what are the components of sperm? Let us discuss it. See, it is the structure of the sperm. If you see clearly, it is the head part. Three parts will be present. One is head, second one is neck, third one is the tail region. It is the complete tail region. Tail region is again divided into middle piece, principal piece, up to here. It is the principal piece and end piece. Okay. Keep it in mind. If we talk about head, head consists of head cap. Head cap will be present and acrosome will be present. Nucleus will be present. Okay. Head cap, acrosome, and nucleus. See, if we talk about acrosome, it contains hydrolytic enzymes. Hydrolytic enzymes. The enzymatic part of sperm is acrosome. It contains acrosin, which cleaves the zona pellucida region of ovum. Okay. Next, it also contains hyaluronic acid, hyaluronidase, hyaluronidase, an enzyme which plays a prominent role in cleaving the coronary radiata, which is a layer of ovum. Okay. These two combinedly called as sperm lysins because it helps during the time of the fertilization. Okay. Next, along with this, acid phosphatase will be present, proteases will be present, which plays their specific function during the penetration of the ovum. Okay. Next, if you talk about the neck, it is the proximal centriole. Here, it is the proximal centriole. I will show you clearly. This one is the proximal centriole, and here is the distal centriole. Okay. It is PC, it is DC. PC has the specific function which plays prominent role during the time of the cleavage because it produces the spindle system which promotes the initiation and elongation of the spindle fibers, which is plays a major role in movement of the chromosomes during the respective stages. Okay. Next, distal centriole. Distal centriole produces axial filament. Okay. Now, middle piece. Middle piece contains mitochondrial sheath, Nebenket sheath, and at the posterior there is one sheath, one ring. That means a ring. See here the ring is. It is the ring. That ring is called as ring centriole or annulus or see Jensen's ring. Annulus or Jensen's ring. Okay. Next, it is the axial filament which continue to the principal piece and from there to end piece. Keep it in mind, there will be plasma membrane. See, there will be plasma membrane that covers the entire structure of the sperm except the region of end piece. Only in end piece there will be no plasma membrane. Remaining on other all structures is covered by the plasma membrane. Okay. Next, if you talk about the ODF, outer dense proteins or fibrins, peritubules and central tubules. Based on this, there will be one prominent question. So I discussing that also. See, 1 minute. See. See, axoneme will be present. In central axoneme, there will be inner central tubes. Cover around that there will be peritubules. Okay. Already I informed that. Around the axoneme there will be outer dense fibers. From inside to outside, it is the arrangement. That means inner central tubules and beyond that peripheral tubules, beyond that outer dense fibers will be present or proteins will be present. Okay. Now, based on the arrangement, at a specific part of the tail, the number varies. Let us see that. Okay. See, it is the tail from here to here, here. From here to here. It is the tail region. Already I informed that it is divided into three: middle piece, principal piece, and end piece. See, in mid piece, it is the central. See, central tubules, two is present. Next, these are the peripheral tubules. These are the peripheral tubules. That means doublets. How many peripheral tubules? 9 pairs. Surrounding that, there is 1, 2, 3, 4, 5, 6, 7, 8, 9. ODF. That means outer dense fibers or proteins. Outer dense proteins. It is 9 in number. When we come into the principal piece and if we check that, see, central tubules will be 2 and the peripheral tubules will be 9 pairs. That means 9 doublets. If we talk about the ODF, see, 1, 2, 3, 4, 5, 6, 7. Only 7 is present. Remaining 2 is replaced by the, see, this one. Replaced by some structures. Okay. Next, if it come near to the end piece and if you check that one, central tubule is 2 will be present. 2 will be present. And if you talk about the peripheral tubules, it will of 9 doublets. If you talk about the ODF, there is no ODF. That means the number that is varying in these three, that means CT, PT, and ODF, in these three, which one is varying from the place of tile means that ODF. That means outer dense fiber is fiber. The number of outer dense fibers is varying from the part of each tile. From one tile part to another part of the tile, the ODF is varying. So we need to keep in memory that each part of tile has how many number of ODFs. Okay. See, middle piece has 9 ODF, whereas principal piece has 7 ODF and the end piece has 0 ODF. It is mentioned clearly here. See here. Middle piece 9 will be present, peripheral tube 9 will be present, central tubes 9 will be. It is same. These peripheral and central tubules both are same. So just we need to know how many. How many peripheral tubules means 9. Doublets will be present. How many central tubules present in tail means just simply wherever it may be. It is in a mid piece or principal piece or end piece. The number will be 2. Okay. But when we are talking about ODF or ODP, then we need to take care that middle piece contain 9 and principal piece contains 7 and end piece contains 0. Here, 2 types of question will be arise. First question, number of ODFs or ODP present. And another type of question is number of ODF or ODP replaced by longitudinal columns of fibrous sheet. This replaced by longitudinal columns of fibrous sheet. Keep it in mind. Replace how many? If we talking about the ODF or ODP present means then it will be the answer will be 7. Where in principal piece. If you're talking about the how many is replaced, then 2. These are the numbers for principal piece. But when we come near to the end piece, number of ODFs 0. Number of ODFs replaced 0. So the main question that came from the principal piece. So we need to be careful while dealing with principal piece in terms of number of ODFs. That means outer dense fibers. Okay, that is the structure of spermatozoa or sperm. Next, factors affecting spermatogenesis. See, very important topic. Okay, factors affecting spermatogenesis. Basically, spermatogenesis is affected by both environmental and physiological. That means if we talk about the environmental, the temperature, scrotal reflex, others like deficiency, consumption of alcohol, all these things come under environmental factors. Let us discuss each one. Temperature. See, normal body temperature are about it. See, normal body temperature for spermatogenesis is less than 2 to 2.5 degrees Celsius than the body temperature. Normal body temperature. Okay. Above it, spermatogenesis will be not happen. Below this, that time also the spermatogenesis will be inhibited. Okay, keep it in mind. Optimal temperature is 2 to 2.5 degrees Celsius below the body temperature. Always it is maintained by the scrotum due to the presence of hydrocele that is present in between the two layers of tunica vaginalis. Already I discussed in the starting slide. Okay. Next, scrotal reflex. Already I mentioned the elevation of testis and elevation of scrotum. In these two functions, two types of muscles will come, and those muscles are the very much important muscles that cause the elevation. Nothing but scrotal reflex. Here that is mentioned. Cremaster muscles and Daughter's smooth muscles in scrotum. It plays a major role in creating the scrotal reflex. That means position its towards or away from the heat of the bone. If any changes happen in the scrotal reflex, then if there is a need for movement of this scrotum towards the testes, but it is not happening, then surely there will be less temperature than the required. Thus, the spermiogenesis is not promoted. Okay, like that, the scrotal reflex also reflect the spermatogenesis. Next, others, that means deficiency of vitamin E, B, A, and elements like Zn plus 2. Okay, all these things will be cause the effect, so its effects on spermatogenesis. Next, infectious disease, syphilis, gonorrhea, all those things comes under the infectious disease. AIDS, even those are sexual disease, of course, they also cause the effects on the spermatogenesis. Next, if we talk about the X-ray exposure, if continuously there is a scanning over the testes, it causes the mutations in those cells, and what are the essential proteins that need to be present during the time of the spermatogenesis, those are mutated and cause the effect on spermatogenesis. Okay. Next, metals like lead, cadmium, dioxin, alcohol consumption, pesticides, anabolic steroids, all these things also reflects the spermatogenesis. Okay. Next, oxidative stress also cause the DNA damage. So when DNA is damaged, the proteins that need to be come from the gene will be not translated or not produced during the time of transcription and translation. So the spermiogenesis, the signals present required for the spermiogenesis is not generated. Thus, the spermatogenesis is restricted. Okay, these are the environmental factors. Next, if we talk about the physiological, now we are talking about the physiological. See, in physiological means that is metabolical. How the metabolically? Because of hormonal actions, hormonal mechanism. Hormones, that means endocrine. Okay, keep it in mind, endocrine. See, not only endocrine, there is paracrine also. Whenever time permits me, surely I will say how the paracrine function is involved in the control of this spermatogenesis. Okay, see, hormonal mechanism. When puberty attains, the central nervous system gives signals towards the hypothalamus. For every 90 minutes, hypothalamus secretes gonadotropin releasing hormone. Gonadotropin releasing hormone. Okay. Next, see, keep it in mind. HPG axis plays a major role in hormonal axis. H stands for hypothalamus. P stands for pituitary. G stands for gonads. These three structures plays a prominent role in the hormonal mechanism. Now see, hypothalamus releases the gonadotropin releasing hormone, and it reaches the adenohypophysis of pituitary by the means of hypophyseal portal system. By this track or by this system, the gonadotropin is reached into the adenohypophysis, and now this adenohypophysis by the blood entering into the blood, it the adenohypophysis secretes the FSH and LH. That means interstitial cell secreting cells. These are the gonadotrophins, which has peculiar function over spermatogenesis. See, if we talk about the FSH, it enters into the blood and reaches the Sertoli cells, which is somatic cells of seminiferous epithelium, and cause the promotion of spermiogenesis, release of inhibin, and release of androgen binding protein. Okay. Next, It also release from adenohypophysis. Luteinizing hormone is also released, and by the means of blood, it reach the Leydig cells, which are the interstitial cells. Interstitial cells, okay, which present in intertubular space. Intertubular space. Okay. Now these Leydig cells produce androgens. The chief androgens is testosterone. Okay. Now this testosterone, by binding to the androgen binding protein, thus enters into the spermatogonium and thus promotes the spermatogenesis. Okay. Keep it in mind. In brief way, I will give you clear cut. At puberty, central nervous system gives signals to the hypothalamus to secrete the gonadotropin releasing hormone. For every 90 minutes, there is a release of gonadotropin releasing hormone. And this gonadotropin releasing hormone, by the means of hypophyseal portal system, it reaches the adenohypophysis, that means anterior pituitary. Okay. Next, anterior pituitary secretes the gonadotrophins, which is nothing but FSH and LH. Each one has specific role in the spermatogenesis. FSH stimulates the Sertoli cells, and Sertoli cells cause the secretion of Spermiogenesis, inhibin, ABP, estrogen, etc. And few more Leydig cells stimulating factors. All these things will be released by the Sertoli cells. Now, the Leydig cells is present in the interstitial space, intertubular space, will be innervated, that means stimulated by this LH, and cause the release of androgens. The chief androgen is this testosterone, which plays a prominent role in few physiological functions. One of that is spermatogenesis. How it will enter into the seminiferous tubules means that it binds to the androgen binding protein that released by the Sertoli cells into the lumen. Not lumen, into the lumen, and from where lumen to intertubular space. Intertubular space. Okay. Now this testosterone binds with ABP and reaches the spermatogonium. Spermatogonium, and thus promotes the spermatogenesis. This is the pathway. Okay. See, I will. It is the pathway. Here, a clear view is given. I will explain it. See, first one. It is here the hypothalamus will be present. Whenever there is signal from the central nervous system, hypothalamus release two types of hormones. One is releasing hormones, and the second one is inhibiting hormones, based on the signal that it got from the central nervous system. Okay. See, if releasing factors, releasing hormones. See, whenever hypothalamus release gonadotropin releasing hormone, see here, gonadotropin releasing hormone. Okay. It reaches the anterior pituitary by the means of hypothalamic hypophyseal portal system or tract. By means of that, it reaches from hypothalamus to anterior pituitary, where this gonadotropin releasing hormone stimulates the anterior pituitary and cause the release of gonadotrophins. Those two gonadotrophins are LH. Here the LH. See, one is LH and second one is FSH. LH, first I talking about the LH. LH shows its effect on the Leydig cells. And one more thing, very, very important one is, till now I thought that only gonadotrophins are released from the pituitary gland. That means anterior pituitary gland, because of release of gonadotropin releasing hormone. Not only that, the hypothalamus, when it gets the signal from central nervous system, one more hormone, that is other than the gonadotropin releasing hormone, prolactin releasing hormone is also released. Prolactin releasing hormone. When prolactin releasing hormone reach the anterior pituitary, thus the anterior pituitary, very, very important one, anterior pituitary releases small amount of prolactin. Very, very important. Prolactin. Okay. Now, first I will talk about LH, then prolactin, then FSH. Okay. See, LH, it shows its activity on Leydig cells that are present in the intertubular space. Okay. Now the Leydig cells produce androgens. Chief androgen are testosterone. Testosterone. Now, what are the functions of the testosterone? It shows its activity on Sertoli cells, Second one, spermatogonia cells. Third one, um, secondary sex organs. Secondary sex organs. During the sex differentiation. And also after puberty. Okay, keep it in mind. First one is it shows its activity. See, here the testosterone is releasing. See, this is testosterone. Testosterone released, and it shows its activity on the sexual cells. These are the sexual cells. Wait, I will mark it. These are the sexual cells. This one is the sexual cell. Okay. See, the testosterone that is released by the LC reaches the sexual cells. Okay, and it also reaching the spermatogonium. That means here see germ cells. These are the germ cells. That means testosterone also showing its effects on testosterone. Other than that, testosterone also showing its role on prostate, seminal vesicles, epididymis, ductus deferens, penis, scrotum, etc. Okay. See, here one question is arised based on the timing of release of testosterone. See, generally testosterone is produced in the 7th week of gestation, but seminiferous tubule is formed just before the puberty. PGC changes into spermatogonia happens just before the puberty. That means these two cells, Sertoli cells will be present initially, of course. The seminiferous tubule and spermatogonia, second one and seminiferous tubules will form during just before the puberty. But the secretion of testosterone is seen in the 7th week of gestation itself. Then what the function of this testosterone during this period of after 7th week to before puberty? During this period, what the function of testosterone means? Mainly, it is involving in sex differentiation in early embryonic stage itself. How it will be, I will discuss later. Okay. Now see about the already testosterone. I completed about the testosterone. Now come to the second FSH. See here the FSH is present. See, it is released by the and reaches the Sertoli cells. Now the Sertoli cells releases an ABP androgen binding protein and inhibins. Okay, inhibin. See, one is inhibin. Sertoli cells release inhibin, androgen binding proteins. Okay, these are the next estrogen. See here, estrogen, Leydig cell stimulatory factor LCSF. That means whenever FSH, central nervous system given signal to hypothalamus, hypothalamus will release gonadotropin releasing hormone, which reaches the anterior pituitary. Anterior pituitary release FSH along with LH. But I talking only about the FSH. FSH stimulates the Sertoli cells. Now Sertoli cells produces one inhibin, two ABP androgen binding proteins, three estrogen, four Leydig cell stimulatory factor. During the early stage, that means during the at the time of the 7th week of gestation, these Sertoli cells also produce two. Which one is not two, only one. Mullerian inhibiting substance or anti-Mullerian hormone. But it is not due to the function of this FSH. Because why? I already mentioned at the time of puberty only, the central nervous system will give the signal to the hypothalamus. It is not giving a signal to hypothalamus before the puberty. That means MIS and IMH, which is secreted by the Sertoli cells, is not the product of signal that is obtained after puberty. Okay, it is the main thing. Keep it in mind. Next, see, if you talk about each one, inhibin. Now see, inhibin, first one is there. Inhibin causes the inhibitory role. That means gives the negative feedback mechanism to the FSH. It acts on the anterior pituitary, or indirectly it acts on the hypothalamus. See, indirectly it acts. Directly it acts, see, inhibin directly acts on anterior pituitary, or indirectly acts on hypothalamus. Hypothalamus. If directly acts on anterior pituitary, it causes the inhibition of release of gonadotrophin, especially FSH. Thus, the concentration of FSH is inhibited by this inhibin. Keep it in mind, not every hormone that is released by the gonads is steroidal hormones, because inhibin is the peptide hormone, protein hormone. The hormone which is secreted by the sexual cells, that means somatic cells of the gonads, especially seminal epithelium. These sexual cells are the somatic cells of seminal epithelium. Okay, even though these produce the protein hormone. Okay. Next, if it acts indirectly on the hypothalamus, then it shows its effects on the gonadotrophin releasing hormone. Thus, the gonadotrophin is inhibited. Okay. It is the two pathways how the inhibin control the FSH. Okay. Next, if we talk about the ABP, see, ABP here, the ABP is present. This ABP binds with the testosterone and takes this testosterone, which is present in the intertubular space, into spermatogonium or germ cells, and thus promotes this spermatogenesis. Okay, that is it. Next, estosterone and LCSF. These two cause its effects on the LC, that means Leydig cells, to produce more amount of testosterone, that means androgens. Okay, it is the function of the FSH. Next, if you talk about the prolactin, very, very important. Prolactin. See, central nervous system, whenever gives signal to the hypothalamus, there is release of gonadotrophin releasing hormone along with prolactin releasing hormone will be released. Already I said it reaches the anterior pituitary. It also reaches the anterior pituitary. Now, the anterior pituitary produces gonadotrophins, where anterior pituitary with the signal of prolactin releasing hormone, it releases prolactin, lactic hormone, prolactin. Okay. See, already I discussed all those things. That is caused by the gonadotrophins. Now, prolactin. See, whenever we talk about the prolactin, why prolactin is to be released? Because whenever the LH is released, that means luteinizing hormone is released into blood, it need to come and bind to the Leydig cells. Leydig cells. See, it is the, let us take this as the Leydig cell. When whatever the cell surface, there will be receptors for LH. These are the LH receptors. It has two functions. First function, it modifies the signals of LH by increasing the LH receptors. It increases the LH receptors on the Leydig cells. That means prolactin plays a prominent role in regulating modulation of LH binding to role of prolactin in the spermatogenesis. Okay, it is the mechanism. Next, see, hormone cell interaction during spermatogenesis. See, based on the cell type, I will talk one again. See, it is spermatogonia. From spermatogonia, more spermatogonia will be produced. That is mitotic amplification happened. That mitosis, it is under the control of FSH and testosterone. Next, more spermatogonia is converted into primary spermatocyte. It is due to GDNF SCF factors produced by the sexual cells. Next, this primary spermatocytes is converted into secondary spermatocyte due to the reasons of testosterone. Next, secondary spermatocytes is converted into spermatids due to this testosterone. Next, spermatids is converted into spermatozoa, which is under the control of FSH. That means spermiogenesis under the control of FSH. Already I mentioned in the previous slide. Okay. Next, spermatozoa is released as sperm into seminiferous tubules, which is called as permeation. Okay. These all things, that is majorly FSH and LH plays a prominent role. Okay. See, if you talk about the cells, I will enlarge it 1 minute. See, it is the basement membrane of the seminiferous tubule. Basement membrane of seminiferous tubules. That means it is seminiferous tube. See, it is Sertoli cell. It is Sertoli cell. Okay, it is nucleus of the Sertoli cell. It is the somatic cell. Somatic cell of seminiferous epithelium. Okay, now see, these are the Leydig cells. These are the Leydig cells. It is intertubular space. Okay, now the mechanism. See, okay. By the incoming blood, the gonadotrophins comes LH and FSH. LH and FSH. Okay. Now see, LH. LH shows positive effect. That means promoting effect on the Leydig cells. Okay. Now the Leydig cells produce testosterone. Produce testosterone T. Okay. Now T enters out by blood and reach the secondary sex organs and controls secondary sex organ function and also influence the secondary sexual characters. These are the two important functions of testosterone when it passes away from the testes by the means of blood. Okay. Next, another function of testis is it enters into the Sertoli cell. Okay, it enters into the Sertoli cell and converts into estrogen. Okay, testosterone enters into the Sertoli cells and causes the stimulation of this spermiogenesis and spermatogenesis. Okay. On other hand, it also reaches the male germ cell by the means of ABP. By the means of ABP. Okay. Next, this ABP along with testis reach the male germ cells and shows promoting effect and thus cause spermatogenesis. Okay. Now, on other hand, LH and FSH both cause positive effect on male germ cells. Okay. Now, what will be the effect of FSH on the Sertoli cells? Here, the FSH is present, which comes from the incoming blood, reaches the Sertoli cell. See here, this incoming blood reaches the Sertoli cell. From the incoming blood, the FSH reaches the Sertoli cell. Thus, FSH shows the positive effect on the FSH. Sertoli cells, not FSH. See, from the incoming blood, the FSH reaches the Sertoli cells and it shows the positive effect on the Sertoli cells. Okay. Now, the Sertoli cells produce inhibin, androgen binding protein, aromatase 5-alpha aromatase, 5-alpha aromatase. Okay. It is STP, stimulating proteins, STP. Next, see, inhibin, it is released into the blood and which reach the hypothalamus or anterior pituitary and cause the respective inhibiting or negative feedback mechanism. And the ABP, which is released into the out of Sertoli cells. These cells binds with the testosterone and reach the male germ cells and cause the spermatogenesis. Spermatogenesis. Now, 5 alpha aromatase. See, testosterone is converted into dihydroxy testosterone by the means of 5 alpha aromatase. Okay, 5 alpha aromatase. Now, STP. This STP means that stimulating factors. Okay, these reach the Leydig cells and cause the increase of testosterone release from the Leydig cells. This is the endocrinology of the testis. This is the mechanism that involved in the endocrinology of the testis. Okay. Now, sex differentiation. Just one slide. Question here arises is, generally at puberty, central nervous system gives signal to hypothalamus. Thus, hypothalamus releases gonadotropin releasing hormone, which reach the anterior pituitary. Thus, anterior pituitary releases gonadotrophins. These gonadotrophins are FSH and LH. Keep FSH aside. We are supposed to talk about LH. See, now LH reach the Leydig cells present in the intertubular space of testicular lobules. See, these Leydig cells, it shows its effects on Leydig cells and cause the release of androgens. Okay. Now, the chief androgen is testosterone. Chief androgen is testosterone. Now, the function of testosterone is, we all know that. One, it shows functions on Sertoli cells. Second one, it shows on germ cells. Third one, it shows on secondary sex organs. When these all things are happening at the after puberty. But if we see within the 7th week of gestation, the release of androgens, especially testosterone, is noticed. Without the signal of central nervous system, which is occurred at the time of puberty. Okay. Why this testosterone is producing in 7th week itself? What the role of testosterone in gestation period itself? If we talk about that, then a new topic, sex differentiation, will be arise. Just I will give simple view on the sex differentiation related to males, not about females. See, sex differentiation in males primarily involves in fetal stage. Okay. Mechanism. What is the process involved in it? Okay. First, there is XY chromosome. Nothing but genotype. Okay. XY chromosomes on the Y chromosome presence of SRY gene is noticed. That means this XRY gene codes for testis determining factor. Next, it causes the primordial gonads differentiation. Thus, this primordial gonad differentiation causes fetal testis formation. Now, in testis, there will be two types of cells: Sertoli cells and Leydig cells. Now, see, Sertoli cells. If we talk about the Sertoli cells, without having any gonadotrophin, it activated and it produced the anti-Mullerian substance or hormone or Mullerian inhibit substance, okay, which cause the regression of Mullerian duct. Thus cause the inhibition of development of female reproductive system. Next, on other hand, LC, Leydig cells, produce testosterone. Now, testosterone directly causes the differentiation of Wolffian duct, and which leads to the formation of epididymis, vasa efferentia, vasa deferentia, seminal vesicles, etc. On other hand, testis in the presence of 5-alpha reductase, it is converted into dihydroxytestosterone. Okay, which plays a major role in formation of or development of male external genitalia. Example: penis, scrotum, prostate, etc. Thus, the sex differentiation in which testosterone, without having the gonadotrophins, performs their role during the time of 7th week in gestation period. Okay. Next, see, after that, termination of spermatogenesis. Final concept in this spermiogenesis is termination of spermatogenesis. It is happened in two ways: by showing negative feedback on FSH and LH concentration, which because these two hormones are main hormones that involve in the spermatogenesis. So, if they are terminated, then surely the spermatogenesis is restricted. First way, effect on concentration of FSH. See, inhibin, a protein hormone secreted by Sertoli cells, act on the adenohypophysis and inhibit the FSH production. See, inhibin already while discussing the hormonal action on the Sertoli cells, spermatogenesis, in previous slide, I already given direct and indirect pathway created by inhibin hormone during the negative feedback on concentration of FSH. Okay. Okay, I will say again, direct pathway, indirect. See, if indirect pathway, inhibin acts on anterior pituitary and stops the production of gonadotrophins, especially FSH. Thus, the concentration of FSH is directly monitored by inhibin, while termination of spermatogenesis. Next, indirect pathway. In indirect pathway, inhibin acts on the hypothalamus and thus cause suppression in production of gonadotrophin releasing hormone, which concentration is low, then it not act on the anterior pituitary. So gonadotrophins are not secreted. Okay. Thus the mechanism involved when inhibin is act on the anterior pituitary or hypothalamus. Next, second one is effect on concentration of LH. Testosterone is the chief androgens, so negative feedback by two ways: directly acting on the adenohypophysis or indirectly acting on the hypothalamus. Then they can affect the concentration of LH and thus lead to the termination of spermatogenesis. Okay, this is the two ways where there will be negative feedback on the concentration of FSH and LH, and thus the termination of spermatogenesis is noticed. Okay, by this the total concept related to spermatogenesis is completed and it is provided for you. Thank you.